welcome. Welcome to the second Cognitive Neuroscience Symposium. This is our annual symposium. It's a second, but it's really the third edition after our rebranding event, for those of you who still remember. So I'm really excited to welcome you all today, and what a glorious sunny day to have it on. So thank you for all of you who made the effort to come. We have an amazing list of speakers. There'll be a lot of exciting discussions, and there'll be panel discussions as well. So I hope that we have a very inspiring and engaging event. I will, of course, like to acknowledge that we are gathered on the lands of the Wurundjeri people of the Kulin Nation. I'll pay my respects to their elders, past and present, and extend those respects to any Indigenous people here today. And of course, the Indigenous peoples have always been making discoveries and sharing knowledge as we are here today. So we are very lucky to continue that legacy. I forgot to say, I am Marta Garrido. I didn't introduce myself. I am the director of the Cognitive Neuroscience Hub. Natalia Brumley is the deputy director of the center, and we've got Sarah Davey over there. She's the hub administrator. She is running around making sure that we all are happy and fed and organized. So thank you to Sarah for putting together such a smooth event so far. I'm sure it will be for the rest of the time. Okay, so before, as I said, we have lots of speakers today. Some of them will be from the University of Melbourne, and some are external speakers. So I won't spoil the beans, but I'm really, really excited to hear all of the talks. But before we do that, I just want to reflect upon some of the achievements that we've had as a hub, as a collective. We did a lot, and this is just a snapshot of a bit that we did. We did a lot more, and also some of what we were able to capture. So we had 14 seminars and talks hosted just this year, so up to now, not including today, of course. And we had 4 PhD completions, so this is a massive achievement for our students, and also for the supervisors and the lab, the other lab members, who often provide a lot of support and good morale and source of coffees and so forth. So if anyone is here who has finished their PhDs, put your hand up. No. They all too tired. They're just at home, just recovering. Okay, but anyway, amazing. We had 4 grants as well awarded, so that is fantastic. Many more that have been submitted. That also is a massive effort, and it will be. I'm sure some of those will be eventually up, as these things often go. And 29 publications. So that is a lot of knowledge put out there. So that's amazing. And as I said, there's more. I'm sure there's more that we weren't able to capture, and we're not even capturing the preprints on the publications here. So there's a lot of work, a lot of effort, and massive achievement for all of us as a collective. And I think we should all be very, very proud of ourselves. Okay, so I did forget to do a bit of housekeeping. So toilets are just around the corner over there. There'll be some water as well. And when it comes to it, we'll have morning tea and lunch that's going to be served downstairs in the Hub Cafe. But Iroaz and Sarah will remind us when it comes to it. So I will now pass on to Iroaz de Monteil. She is a professor in our school, and she will be chairing the next session. Thank you.
Hi everyone, welcome. I'm very happy to be introducing the first session today, which is about the disordered brain. We'll have 3 speakers. And each have 20 minutes to talk, and we'll have questions at the end. So remember your question. It's a test of your memory, and make sure you ask them at the end of the session. So first up, we have Dr. Kelsey Perrykkad. She's a postdoctoral research fellow at the Center for Women's and Children's Mental Health within the psychiatry department at Monash University. Her research focuses on how the brain builds and maintains models of the self by interaction with the environment. She uses theoretical and empirical tools, including predictive processing frameworks, to interrogate questions about self-cognition, agency, mental health, and the interplay between perception and action. And she's also expanded into perinatal mental health research, leveraging longitudinal data sets to explore the links between maternal well-being and child outcomes. And Dr. Perrykkad won the Australasian Cognitive Neuroscience Society's Emerging Researcher Award in 2022 for her work on self-models and predictive processing. So Kelsey, welcome, and over to you.
Thank you. All right. Can everyone hear me? Yep. So, hi, I'm Kelsey Perrykkad. Thank you for the beautiful introduction and the opportunity to speak today. I've actually moved from the Department of Psychiatry at Monash to the Monash Center for Consciousness and Contemplative Studies in July this year, so I've come from Monash. Today I wanted to talk about disordered brain, disordered self. So is there a way we can conceive of the sense of self as being disordered in different psychiatric conditions is kind of going to be the scaffold of this talk. So why might we think there are disordered selves? If you look at the ICD in particular, but also the DSM, some psychiatric conditions are defined by differences in sense of self and self-processing. So for example, borderline personality disorder in the ICD is defined by problems in functioning in aspects of the self, such as identity, self-worth, accuracy of the self-view, self-direction, instability of personal relationships, self-image, identity disturbance. Similarly, schizophrenia is another one we could think of as defined by maybe a disordered self. The ICD description says that it's characterized by disturbances in self-experience, including the experience that one's feelings, impulses, thoughts, or behavior are under control of oneself or an external force. So you might think of things like borderline personality disorder and schizophrenia as kind of self-disorders or disorders involving self-related symptoms, whereas things like depressive disorders, anxiety, autism don't have the self explicitly mentioned in their ICD description or their DSM description. So you might think of those as disorders that don't involve the self as a first pass. So in this study that I'm going to talk about now, to begin with, we take a multidimensional view of the self or of sense of self that captures kind of a range of cognitive processes that help us build and maintain our sense of self, including our sense of agency. So when we interact with the world, do we feel ownership or agency over the outcomes that we receive back from the world? Things like self-esteem, autobiographical memory. So it involves lots of cognitive processes that all come together to build this sense of self. And the question for this study was, how should we understand the relationship between each of these dimensions and psychiatric traits? So are there some conditions that are particularly selfy and others that are not? Or is the sense of self something that's kind of transdiagnostically related to psychiatric disorders? So here are two kind of conceptual pictures. The first that could come out from the research. The first is that there's this kind of psychiatric binary. So on any particular dimension of the self, you might get some conditions whose traits are strongly correlated with that dimension, and other traits, other conditions that are not as correlated with that dimension. Or we might end up with kind of a psychiatric spectrum where some conditions have traits that are very strongly correlated with a particular dimension of the self, and other conditions have correlations, but they're less strong. And so you might think of that as how it kind of comes out. So this study kind of looks at that. And what we found is that all of the conditions we studied, which in this experiment was borderline personality disorders, schizophrenia, depression, anxiety, and autism, we found that all of these conditions, their traits, moderately to strongly correlate with self-concept clarity, which is a particular dimension of the self that we looked at, and explicit senses of self accounted for 17 to 47% of the variance in psychiatric trait variability. What we did, we looked at pairwise comparisons between the strength of these correlations and found that borderline personality disorder traits were more strongly correlated with self-concept clarity than depression, anxiety, and autism. But schizophrenia was only more strongly correlated than autism, not depression or anxiety. So this kind of gives us a picture of that kind of latter case of my conceptual options, where all of the conditions that we looked at least in this study, were related to this dimension of the sense of self, even though some were defined by self-related processes and some were not. And this kind of transdiagnostic view of the self is consistent with some frameworks looking at different ways of categorizing psychiatric disorders. So for example, the RDoC framework, which tries to categorize mental health according to different domains that range from genetic to kind of self-report levels of analysis. And the RDoC framework has self-related processing, agency, and ownership in 2 different categories. So our findings are kind of consistent with that way of conceiving of psychiatric disorders. So I'm going to take a minute to zoom in on which dimension in particular for this study correlated with those psychiatric traits across the board. And that dimension was self-concept clarity. So if self-concept clarity is measured through a questionnaire, here are the items if you're interested in the particulars. But from a broad perspective, self-concept clarity captures the perceived clarity, consistency, and stability with which one perceives their self-concept. So does someone think that their self-concept is clearly defined, internally logically consistent, so it doesn't contain elements that are at odds with one another, and temporally stable? Note that these are structural features of the self-concept, not the content of the self-concept. So it's not looking at whether you're a kind person, you feel like you're a kind person, or that you're a mother, or that you have particular goals and values, all the kinds of things that you might, personality traits, things that you might make up your sense of self. It's more about how all of those elements hang together and how they're perceived to hang together. So if we're looking at the cognitive processes then, we can think about how do we construct and maintain a clear, consistent, stable sense of self. And this is where I think like a bit of a predictive processing story comes in. We can create and construct and maintain a clear, consistent, stable sense of self by resolving inconsistencies and uncertainties with regard to our sense of self. So let me take a practical example. We instantiate who we are through actions we choose. So for example, if I think that I'm a gardener, a good gardener, a kind person, an academic, I might do things that in the world that instantiate the fact that I'm a gardener. I might plant some plants, I feed the homeless, that instantiates the fact that I think I'm kind. It gives me evidence that I think I'm kind. I like sit at a desk and read some papers. This is kind of an academic thing to do. And we know if we're right about those facts of ourselves based on whether or not we get the outcomes we expect. So if we draw a little picture of the internal world and your external world divided by a sensory veil, you only get information about the external world based on the actions you choose and the sensory input that you get back from it. If we take a particular thing that I think about myself, I think I'm a good gardener, this might cause me to go out and buy a pot plant. And I start doing some things with the pot plant. I'm watering it, and I put some soil in it, put a seed in there. The action of planting the plant then causes some things in the world to change that I may or may not have access to. And I can infer what's happening in the world, but I don't have direct information about it. But I do get some information from my senses. So for example, the plant grows, and I get this beautiful flower. And because that plant grew and I get a beautiful flower, that kind of matches up with my expectations of being a good gardener. So it reinforces my self-model. I don't have much uncertainty or inconsistency in what feedback I'm getting from the world and the kind of self I'm putting out there. But what happens if the plant dies and I don't get the kind of feedback from the world that I expect from my actions, either in the short term or a long term? This causes some prediction error, which then has to feed back into my self-model. And I have 2 options for dealing with this prediction error in terms of resolving inconsistencies and making my self-model more clear. One of which is to change the model. So abandon the fact that I think I'm a good gardener. I've just gotten feedback from the world that I am a black thumb and I should never touch plants again. So I'm not actually a good gardener. That's one option. The other option, and you might choose some options in some contexts and with some feedback, and the other option in other contexts with other feedbacks, or depending on the type of person you are. The other option is to change the actions I choose to try and get the feedback that I expected. So maybe I water the plant differently, or I choose to grow plants, or I do something differently in the world to then change my sensory input so that I don't have to abandon the idea that I'm a good gardener. I just have to change my actions. So we looked at this kind of process of self-clarification and making yourself more consistent in a paradigm called the squares task in 2 studies. So I'm going to show you some results from that. But first I want to do a little demonstration because I think it makes things a bit more fun. So the participant's task in this paradigm is to decide which of the 8 squares on the screen they're controlling with their mouse movements. So this is a particular trial from that experiment. You can see exactly what the participant can see, but you can also see a little white dot in the center of the screen. I don't know if you'll be able to. Anyway, you can see the little white dot. When I play the video, the white dot's going to move and become a line. The direction of the line is the direction of the mouse movement, and the length of the line is the speed of the mouse movement. So that just gives you some information about what the participant's doing, because you're not actually moving the mouse in this experiment. But you can play along and figure out which of the squares you think is following those mouse movements. Is there, yeah. So moving to the left, to the left, to the right, to the left, right, up, down, left, right. All right, so that's the trial. It's quite quick when you're watching it as an audience member, but when you're doing it, it's much easier because you're actually moving the mouse. You can test your hypotheses. So I'll just play it again. Left, right, left, right, left, right, up, down, left, right. Any guesses on which square the participant was controlling? Orange? Yeah, that's right. Yeah. So orange is the correct answer. So here's the underlying mechanics of our experiment. So in this video, I'm going to show you what was happening underneath all of that. So you'll see all the same things you saw before. Wherever the participant's moving their mouse, one of the squares, the orange square, is roughly following that mouse movement with a little bit of variability added to each frame. And the variability is in the top left-hand corner. The actual number I can explain if you're interested, but it goes up and down over the trial too, so that's variability and volatility. Then the distractor squares are offset by angle from the mouse movement, and they also turn, they change angle 4 times during the trial when the participant's not turning the mouse. So they do have some behaviors that the participant can't control at all. So you can't just motor adapt and think one of the other ones is you. You should be able to get some information that says that you're wrong about that. The white boxes are the eye tracking estimation of where the eyes are. And then the green box around a square at any given moment is my algorithm's detection of which one it thinks you're looking at. So if we look at what was happening in that trial for this participant, they're kind of flitting between these distractor squares, tending to go when they change to big numbers to things that are smaller numbers, which means that they're closer to the mouse movement. And you can see they're kind of looking all around the screen as they move. And then they finally, by the end of the trial, do land on the correct square. So you get lots of information from a trial like this. Yep.
Were they seeing mouse trails, or it was just the actual mouse itself?
They didn't see anything. They didn't see the mouse itself. Their hand was in a box that covered up their actual.
Yeah, but I mean, mouse trails are like little arrows that follow where the mouse has been.
That
sort of indicates what you've just done.
They don't see any of that. Yep. So they just see the squares on the black screen. They don't see the little white thing. They don't see a physical mouse. Their hand is in a box that's covered up, so they can't look at their hand as they're moving. It's just based on how they think they're moving. Yep. And so if we map this back to our conceptual picture, we can infer from the data that we get what the participant's self-model is, what their hypothesis is at any moment in time about which square they're controlling. So we take that as which box their eyes are looking at. Then you get some information about the actions that they're doing. You get all of their mouse movements. We know the hidden state. We know which square is actually under their control for the most part. And they get some sensory input, which may or may not be from the actual square. So whichever square they're looking at moves in a certain way. And so they can kind of do that process of figuring out which square they're controlling and get better and better over time. And one nice thing is that we can measure prediction error in this experiment. And I can again talk you through how we measure it later. And then we can plot that using inspired by kind of ERP analysis, actually. So we time lock the prediction error measurement, which we get on each frame, to particular actions of the participant and look at how the prediction error changes before and after that action. So in these two experiments, we've got prediction error on the y-axis, time on the x-axis, and then they're time locked to a particular participant action. In the top experiment, the action is them changing the hypothesis about which square they control. So it's when they look from one square to a different square. And in the second experiment, it's when they change something about their actions in the world. So in this case, they're choosing a different environment. And one striking thing, I think, in these two experiments is that we see the same pattern of results across both, where these actions seem to happen at a peak in prediction error. So prediction error is rising, and then they act to reduce the uncertainty that they're getting about what they control, and then it falls. The falling is less interesting, the rising is the interesting part. But also that action and the peak, the size of the peak of prediction error at the time of the action depends on the participants, or is associated with the participants' autism quotient score. So participants with lower autism traits, fewer autism traits, tolerate more prediction error before they choose to act to reduce uncertainty. Participants with higher autism traits, so the darker blue lines, are choosing to act at a lower level of prediction error. Okay. And so what we find from this study is that these two studies, individuals with more autistic traits choose to act earlier when they have prediction error about what they can control. That is, sensations that are attributable to themselves in these experiments. So if we link this back to the conceptual question, clarity and consistency are accounted for by inference and hypothesis testing, which is done differently depending on neurodiverse traits, is what I wanted you to get from those experiments. But what about stability? So if selves are inferred based on environment-agent interaction, selves must be subject to learning processes, classic cognitive learning processes, that reflect when these contingencies change. So if we're looking at the stability, which instability seem to be associated with psychiatric traits, the right kind of instability must not be disordered. Learning is instability involves learning. Learning is not always disordered. What happens in a case of healthy self-transformation? So in this study I'm going to show you next, we were looking at pregnancy as an example of healthy self-transformation. So I'm running out of time, so I'm going to run through this pretty quickly. The idea is that pregnancy is a canonical transformative experience that changes who you are across a whole bunch of levels of the cognitive hierarchy and across multiple time scales that span during pregnancy and after pregnancy when you're taking on the role of mother. And this could either precify the self model, increasing attention to the sense of self. You're testing the model, you might get thrown out of a local minima. But it could also destabilize the self model. If everything's changing all at the same time, there's nothing to kind of anchor you, that might lead to a lot of the vulnerabilities in mental health during pregnancy and the postpartum. So we really weren't sure what was going to happen to the sense of self during pregnancy. We've looked at this now across two different studies, one in pregnancy and one in the postpartum, and I'll just show you kind of a schematic of our results. What we found in the pregnancy study was that compared to never been pregnant women in the first trimester experience an increased self-reported sense of negative agency, so they perceive that they're out of control more. They have a learning deficit, which we measure in a self-related learning task, but the learning deficit is not specific to the sense of self in the findings. In the second trimester, we find an increased self-reported self-efficacy. People feel more competent they feel a sense of competence and a belief that they can achieve their goals across environments. Also, they have an increased self-reported body agency, so they feel more attractive, feminine, pride, competence, confidence, and you get a recovery of that learning to baseline. So you seem to have a stronger sense of self in the second trimester. And then by the third trimester, a lot of those effects kind of disappear, and you get that learning deficit back in preparation for birth. Across the whole pregnancy period, we have increased feelings of body estrangement and body visibility, so you feel like your body is touched, stared at more in public. And in the postpartum, new results, subject to change, but we find some increased self-reported body estrangement, which suggests some diminishing of the self-concept or self-related processes in the postpartum. So there does seem to be some shifts in across a range of different cognitive processes related to self in pregnancy and the postpartum. But interestingly, we did measure self-concept clarity in these two studies and found evidence for no differences in self-concept clarity, which implies that perceived clarity, consistency, and stability of self-concept appears unchanged in the transition to parenthood, even if other aspects of self-processing are changing, so body representation and learning. So, in conclusion, what are my take-home messages? I'm not sure I've answered the question of can a self be disordered in what ways, but hopefully I've given you a little bit of information about how you might think about that question. First, I said that traits of disorders are transdiagnostically related to selves perceived as unstable, unclear, inconsistent. I said that we act to clarify the nature of ourselves, and how we do this is related to neurodiverse traits, so autism traits in particular, in my studies. Perception of stability and not actual stability might be important for healthy selves. But I also wanted to note that the first two conclusions are ultimately based on individual variability in healthy general populations measuring psychiatric traits, not psychiatric disorders. So this suggests that not all cases or levels of instability and ambiguity are going to be disordered, but they still seem to be related to those kinds of disorders. And I just want to thank my co-authors. If you're interested in more of my work, you can follow the QR code.
Thank you very much. Kelsey, that was great. Very interesting paradigm that you've got your squares task. Robin, where's Robin? So next, we have Dr. Robin Cash. Can I let you, do you want to put this on? So Robin is a senior research fellow in brain stimulation in the Department of Biomedical Engineering at the University of Melbourne. He's funded under an NHMRC EL2 and leads the brain stimulation platform here at the university. And his research centers on neuroimaging-guided personalised brain stimulation, in particular TMS, for treatment-resistant mental health conditions such as depression. He has spearheaded methods to tailor TMS targeting by leveraging individual brain connectivity and neural biomarkers rather than relying on fixed skull landmarks, improving the precision and efficacy of stimulation. Dr. Cash is currently leading a large multi-site clinical trial across Melbourne, Sydney, and Brisbane, exploring the clinical benefits of these personalised TMS methods. So welcome, Robin, and looking forward to your talk.
Oh, yeah, cool. Cool. Thanks, everyone. Thank you. So I'm just going to talk about my research, which focuses on personalised brain stimulation. So focusing on the application of personalised brain stimulation in treatment-resistant depression. And this approach aims to optimise the therapeutic outcomes of transcranial magnetic stimulation. Is it not working? Oh, hi. How about that? Oh, still the same. Oh, yeah, that's better. Oh, yeah, it is too. Yeah, so this approach aims to optimize the therapeutic outcomes of transcranial magnetic stimulation by aligning brain stimulation targets with person-specific brain network architecture. So TMS, who's actually heard of transcranial magnetic stimulation? Okay, that's good. So it's a brain stimulation method that's used in research, but also in clinical therapy. And the most common therapeutic application of TMS is for treatment-resistant depression, where it is typically targeted to the left dorsolateral prefrontal cortex. And you can see here, this is a typical TMS pulse sequence. So it involves around 25 minutes of patterned bursts of TMS, high-frequency TMS. And this pattern of stimulation is based on cellular paradigms where similar patterns of stimulation result in neuroplastic changes. In humans, over the course of several weeks of daily TMS therapy, some people will respond very well, but many do not. And overall, the response rate is around 35 to 50%. And this is not bad considering this is a treatment-resistant population, but nonetheless leaves a lot of room for improvement. One basis for this variation could be clinical or demographic factors. Although these have shown somewhat weak and inconsistent effects across studies. Another potential factor is suboptimal spatial targeting. But why do we think that that could matter? So here's a simple analogy from the motor cortex, where the importance of targeting is immediately evident during brain stimulation. So we know that different muscles have distinct, maybe I've got some kind of timer on this. But different muscles have distinct representations. So for example, if we stimulate the hand representation, this will elicit a muscle twitch in the hand muscle. Whereas if we stimulate the leg area, this elicits a movement in the leg. So in terms of targeting, we obviously wouldn't stimulate the leg area if our target muscle is the hand muscle. And similarly, if we deliver repetitive TMS to target a hand muscle, the plastic effects will be maximal at the hand muscle. So the enduring effects of stimulation will be maximal at the hand muscle and taper off at other muscle representations with increasing distance from the TMS coil. So this is very simple in the motor cortex. But the problem is it's much less straightforward to determine an optimal target for a psychiatric disorder. And over the past 2 decades, a range of targeting methodologies have been explored for depression. And you can see here that these have targeted quite different subregions of the dorsolateral prefrontal cortex, moving from left to right, scalp-based targeting techniques. It's almost like it follows my voice. Scalp-based targeting techniques through to like EEG-based coil placement, and then a whole range of different MRI-based or imaging-based targeting techniques. So just to give you an overview, conventional TMS targeting, which is still used in most clinics around the world, uses scalp-based heuristics. So this is something like figuring out where the motor cortex is, so a region that elicits where a TMS pulse elicits a muscle twitch in the hand, and then just shifting the coil 5 centimeters forward. So it's very rough, very crude. This is basically computed using a measuring tape, and it offers the advantages of speed and simplicity, but results in substantial spatial variation both within and between methodologies. So this figure depicts real data from clinical trials where each dot represents the stimulation site for one patient. So you can see, you can see a lot of spatial scatter across patients for a given method as well as between methods. So this spatial scatter may actually result in different subsets of brain networks being preferentially targeted across different patients and targeting techniques. And in addition, you can see here that the functional architecture or spatial mapping of these networks actually differs from one person to the next. And it's now thought that this inconsistent targeting might contribute to variation in TMS therapeutic outcomes. So based on this spatial scatter, people have explored whether it might be possible to make TMS clinical outcomes more consistent by using neuronavigation to ensure that the DLPFC is consistently targeted. So this is neuronavigation. Basically, a person undergoes an MRI, and then the MRI image is uploaded to a neuronavigation device. The head in 3D space is co-localized with that MRI, as is the TMS coil, and then you can use the neuronavigation to precisely position the TMS coil on exactly the brain region that you want. There's also a robotic form of this, so either a person does it or a robot can now do it. In the context of depression, early studies used neuronavigation to target a single one-site-fits-all anatomical coordinate across all individuals. However, the results of these studies were unconvincing, perhaps in part because the optimal target has remained unknown. And another factor may be that a single anatomically defined target may not appropriately account for the functional diversity across individuals. More recently, brain connectivity-based targeting strategies, and in particular targeting strategies that are informed by an individual's brain connectivity or functional connectivity, have shown a robust relation to TMS therapeutic outcomes, but which brain connection should we target? So in the context of major depressive disorder, several retrospective studies have shown that TMS treatment outcome relates closely to brain connectivity or functional connectivity between the dorsolateral prefrontal cortex and a stimulation site and a deeper brain region known as the subgenual cingulate cortex. So this provides an obvious opportunity to improve treatment outcomes by prospectively selecting the DLPFC stimulation site or target based on optimal connectivity with the subgenual cingulate cortex. The easiest approach would be then just to identify a single one-site-fits-all DLPFC target site that should work for all individuals, defined by a group average map of functional connectivity averaged across all individuals. Just before we move on, it's worth adding that the initial reason for focusing on the subgenual cingulate cortex is that this region is involved in negative effects, so negative emotions, and has been implicated in antidepressant response across a range of different therapies. On this basis, the subgenual cingulate cortex was also selected as a target region for deep brain stimulation. So a potential disadvantage, though, of this one-site-fits-all targeting approach is that there is substantial inter-individual variation in brain network architecture, particularly within the prefrontal cortex. So this is highlighted here in hot colors, yellow and orange. And this corresponds to increased evolutionary development within this brain region. These areas can vary to such an extent that individuals can be reliably identified from a large sample based on their functional connectivity fingerprint. And critically, as mentioned earlier, a few studies have delivered TMS to a singular one-site-fits-all target site, but haven't shown any compelling improvement in treatment outcomes relative to conventional targeting. So together, these findings provide a strong rationale for personalizing the DLPFC target site and defining this site according to connectivity with the subgenual cingulate cortex. But at the time that we started this research, it was around 2019 when I joined the university, it wasn't clear whether precise personalized targeting was even feasible. And a lot of people were saying it wasn't. For personalized targeting to be based on functional connectivity to be feasible, it is essential to be able to reliably identify personalized targets for a given individual that remains stable and near identical in position across two separate brain scans. But when we started this work, a paper had just been published are using conventional computational methodology, indicating that for any given individual, targets differed by an average of 3.5 to 4 centimeters between two separate scans, even when using the best quality brain scans from the Human Connectome Project. So in other words, personalised targets did not appear to be reproducible, and personalisation did not appear to be feasible. So at that time, we worked on developing more accurate computational personalisation methodology, and this ultimately enabled us to precisely and reliably pinpoint optimal personalised connectivity-based brain stimulation targets. And we verified in 1,000 individuals from the Human Connectome Project that the sites we identified varied by only 2 millimeters or a single brain voxel between scans repeated the next day or even 1 year apart. Importantly, these personalised targets were not only robust, but also differed substantially between individuals, demonstrating that our approach preserved inter-individual variation in brain network architecture. Interestingly, we also had familial data from the Human Connectome Project and found that targets were more similar between monozygotic twins compared to dizygotic twins, non-twin siblings, and unrelated individuals, suggesting that these targets are stable and under genetic control. We then tested, so that's all well and good, but does it have any clinical relevance? So we then tested whether personalised targeting is clinically meaningful in a sample of 26 individuals who had previously undergone a brain scan, who had received TMS for depression, and for whom the stimulation target sites and treatment outcome was known. So we retrospectively computed the optimal stimulation site, personalised stimulation target from each individual's brain scan. And we anticipated that individuals whose actual stimulation site was closer by chance to their optimal stimulation site would have a better treatment outcome. Here you can see the scatter of optimal personalised stimulation target sites across this cohort. And here you can see that our data confirmed this hypothesis. So this figure shows that individuals who serendipitously received TMS closer to their optimal stimulation site consistently had better treatment outcomes. So our findings have since been replicated by several independent teams around the world. And several clinics and research institutes have started to implement our methodology. Our colleagues have also established the Queensland Neurostimulation Centre in Brisbane, which was the first in the world to offer personalised TMS therapy for major depressive disorder. This is now becoming a major thing in the States. And I just had an email this morning from a company that's looking to use our method and have it FDA approved. So this is the clinical pipeline. Someone undergoes an MRI scan for 15 minutes. We then compute the TMS target site, and then the individual undergoes TMS therapy. The results to date from the Queensland Neurostimulation Centre have been promising and have just been published. So they use this cobot, the robot that positions the coil. This is a distribution of target sites. And the key point here is that clinical response and remission rates with personalised targeting were much higher than those seen previously with conventional targeting in patients in RCT, like randomised clinical trial, control trial, like patients, so with more strict, more pure depression. But unfortunately, the clinical efficacy dropped off in tiers 2 and 3. So those are individuals or groups with more comorbidities, more complex clinical presentations. So that's something that we still need to resolve, how to treat those patients. In Australia, we're now running a double-blind multi-site TMS study in 300 people across Brisbane, Melbourne and Sydney to compare personalised targets with scalp-based targeting. We've also had promising findings with our personalised targeting pipeline in 2 small pilot studies run by our collaborators in the United States. And how much time do I actually have? 5 minutes? Yeah. So Another key question is whether the potential benefits of personalisation based on functional connectivity generalise to other brain regions and TMS applications. And in short, we conducted a similar retrospective analysis using our methodology and substituted the depression circuit with this hippocampal parietal memory network in an existing data set. And again, in this data set, we saw that closer proximity between the actual and personalised circuit target, personalised circuit-based targets was associated with better TMS-evoked improvements in memory performance, suggesting that the benefits of personalised targeting generalise beyond just the prefrontal cortex. We also found the same in a substance use disorder TMS dataset, where closer proximity to personalised targets defined by a craving circuit was associated with better clinical outcomes in craving reduction. And further, the advantage of personalised targeting was only evident in the active, but not in the sham group. Other teams have seen the same advantages of personalised targeting in autism, as well as post-stroke aphasia. And symptom-specific targets have also, there's also some research looking at symptom-specific targets now. So in our previous work, we showed that many, but not all symptoms improve when the TMS target is aligned with functional connectivity to the subgenual cingulate cortex, and more specifically, anxiety and somatic symptoms, so bodily symptoms, tend to improve less. So this has motivated research aimed at delineating new symptom-specific targets or circuits, which allow for symptom-specific TMS targets. In other research, we have delineated brain circuits associated with emotional and cognitive symptoms in unipolar depression, shown here. And in theory, these could provide two separate TMS targets relating to these two different symptom domains. And we can see here that this could potentially be useful. So this figure shows how affective and cognitive symptoms separate out and respond differently at different rates during and following treatment. The same approach has been used to delineate circuits involved in a range of other psychiatric and neurological disorders, and these can then be used to define circuit-based personalised brain stimulation targets. And several of these are now being tested in randomised clinical trials. So with that, I'd just like to thank all of the people listed here who have been involved in this research. Thank you. Yep.
We'll take
Questions at the end.
At the end. So our final speaker for this session is Associate Professor Jacqueline Anderson. Dr. Jacqueline Anderson is an Associate Professor in Clinical Neuropsychology in the Melbourne School of Psychological Sciences. Her research interests primarily relate to outcome after mild traumatic brain injury, and she currently leads a multidisciplinary investigation examining factors contributing to these long-term outcomes. She also leads a study that is investigating a novel treatment program for individuals who have not recovered within the typical time frame. And Associate Professor Anderson also holds an honorary position as Senior Clinical Neuropsychologist at the Alfred Hospital. So looking forward to your talk.
Thanks, everyone. Is that, people hear that? Doesn't sound like it's working. No. I don't know. I haven't got anything higher on me, so I can just use it. Thank you. Or I can yell. Can people hear me before? I don't have to hold a microphone.
Yeah, we can hear you.
Yeah, okay. All right. Maybe I'll just yell. Got that voice. Thank you very much for asking me to give a talk on some aspects of what I've been doing. What I'm going to do today is talk about an aspect of mild traumatic brain injury or concussion recovery that hasn't really received a lot of attention in recent years, or in fact at all. So just to put it into context, I am looking specifically at patients who are in the broader community. So estimate 90% of research in this area is done on elite athletes with sports concussion or with military veterans, purely because they're the groups that have all the big funding bodies. But actually, the majority of individuals who have concussion are just community-dwelling, average people like us who have concussion events and are very, very much under-researched as a group. Before I talk any further, I just want to run through, I'm not going to name all these people, I will be here forever, but I'd particularly like to highlight a few people for this body of work that I'll be talking about today, and that is Professor Mark Seal and Joseph Yang and Richard Baird from MCRI, and then some PhD students and MPsych students, so Lucy Eyre and Maggie Baird, and Lana Hickson, who did this work with me. So just to give you a brief background, it's mild traumatic brain injury or concussion. I'm not going to argue about the definitional differences between those two terms, so I'll just use them interchangeably. It's really, really prevalent within our community. To give you some context, people have concussion more commonly per year in Australia than all people having stroke, for example. So it's highly prevalent in our population. The vast majority of cases, as I said already, reside in the community. They're not elite athletes having sports concussion, and they're not military veterans. Why does it matter? It matters because the mechanism of injury and the recovery profile of patients who are elite athletes or military veterans is very different to the normal population. So the normal population needs to be looked at directly rather than people assuming that the research translates from other groups. Although a lot of attention, in fact, the majority of attention over the last however many decades has focused on moderate to severe traumatic brain injury, in actual fact, when you crunch the numbers, mild traumatic brain injury, due to its prevalence, constitutes a larger financial healthcare burden than moderate to severe injury combined. So it is a major issue in terms of the financial costs, and as I'll talk about, there are individual costs as well to having concussion injuries. It's been established for quite a long time that the majority of patients who have mild traumatic brain injury, so 80% of them, will fully resolve in terms of their objective cognitive function within 6 to 12 weeks. And 12 weeks is being pretty generous. Most people would say you'd be expecting full resolution within the first 4 to 6 weeks, but certainly by 12 weeks post-injury. In more recent years, there's been a shift in attitude towards understanding recovery, however. So there's been a move away from just saying objectively cognition has normalised and therefore they're all fine, to an examination of the personal experience of recovery of patients who have concussion. And that data is not quite so promising. So what it shows is that more than 50% of patients, adult patients I should clarify, continue to report some elevation in their post-concussion symptoms 12 months after injury. Now, there's a big caveat to this, which is that this research has come about from studies that have looked at patients who've admitted, or not admitted, who have presented to ED because of their concern about their concussion. We know that about 50% of people who have concussion don't ever go to hospital, and those people potentially could be milder. So the more than 50% number is probably an exaggeration, an overestimation of the issue, but it is certainly the case that it's considerably more elevated than ongoing objective cognitive deficits after injury. So there is now this situation where there's this disconnect between people believing that there is objective cognitive recovery after mild TBI, but loads and loads of people keep complaining, keep reporting ongoing symptoms. And so there is this tension in the literature about what might be contributing to that higher amount of complaint. Lots of work is being done looking at the psychosocial contribution to outcome. So we know that psychological distress increases risk of elevated ongoing persistent symptoms, female sex, heightened fatigue, approach coping, if you have lower amounts of approach coping style, if you have a stronger belief that your symptoms are directly due to the head injury, not due to something else, you're more likely to have persistent symptoms. And if you have elevations in terms of neurotic personality style, then you're also more likely to have higher risk of poorer of ongoing symptoms. But for a long time, no one has really paid much attention to neuropathology. And the reason for this is because up until pretty recently, our neuroimaging techniques have been too gross to really identify the more subtle kinds of neuropathological impairments that occur following mild traumatic brain injury. And so with our relatively grosser instruments, individuals with mild TBI looked normal, and so the assumption was that neuropathology had recovered. In fact, that view continues to persist so strongly that I had a GP reviewer responding to me yesterday relating to a completely different paper, that it is well understood that there are only functional and transient changes associated with mild traumatic brain injury. And then here's my point. But the point is that clinically, that is a very well-established, well-accepted position that exists out in our clinical community. Neuropathology, structural neuropathology, is not a relevant factor for patients with mild TBI. So let's just talk about what neuropathology of concussion is. Primary neuropathology is something called diffuse axonal injury. It is important to just be aware that irrespective of the nature of the hit or the direction of the hit, the mechanics or physics of the concussional event mean that the brain will move within the skull in two directions. It will move in a linear direction, in the direction of the hit, and it will move in a rotational way, so that the brain will have some rotational aspect to it. Why does this matter? It matters because the rotational aspect of this means that subcortical, central, medial structures are particular points of vulnerability within the brain. It also matters because it means that as the brain is moving, the junction between the grey and white matter, which have different masses, is particularly vulnerable because those two parts of the brain will move at different velocities in the concussional event. The coup and contra coup aspect is, I put up there really because this is like the classic picture associated with traumatic brain injury pathology, and I just want to let people know that actually this is not particularly relevant for mild traumatic brain injury. Coup and contra coup injury is, if anyone's heard of it before in the room, is actually really only relevant to moderate to severe injury. So it is the diffuse axonal injury that is key in concussion. So what I'd like to do is just talk about this study that I've been running for a while. I'm only going to be talking about a subset of the study. It's a much larger study, but most of the neuropathology work has been done on a subset of patients. So about 40 premorbidly healthy adults who've got mild traumatic brain injury. So what happens is we have patients and we have a trauma control group as well. We have patients, they get admitted into the trauma units in Melbourne. They get admitted for physical injury, not for concussional injury. So they get admitted following a traumatic accident for physical injury. When they come up to the trauma ward, my very diligent students go and assess them and diagnose them for mild traumatic brain injury. If they have TBI, they go into the mild TBI group. If they don't have a mild TBI and have had no head strike, they go into a trauma control group. So they are an identical population, one half of which has had a concussion event in addition to their physical injury, and the other half of which have not. Consequently, they are beautifully matched in terms of demographic variables, which I'll talk about in a moment. So they're adults under the age of 65, very consistently with the concussion population, about 2/3 of them are male. They're recruited on the ward, and then we do a whole lot of assessment with them, including structural and functional neuroimaging, around about 9 to 10 weeks post-injury. We do a whole range of cognitive assessment, psychological assessment, including sort of psychiatric features, general psychological aspects, physical things, symptoms, quality of life. Most of that is not relevant to this talk. It's really just the cognition and the neuroimaging stuff that is pertinent. The scanner is a 3T scanner at the Alfred, and everyone was scanned on the same scanner, irrespective of where their initial admission hospital was. So in terms of the group dynamic demographics, in terms of pre-morbid IQ, you name it, they are beautifully matched groups, sex, age, throw me a demographic and they'll be well matched on, basically. Which is really nice because it provides a unique insight into, just the distinguishing feature, which is that one half of them have had a mild TBI and one half of them have not. In terms of injury-related variables, they're really well matched. And of course, only the mild TBI group have the concussion diagnostic criteria variables. So Glasgow Coma Scale score, post-traumatic amnesia, and loss of consciousness. Time since injury at time of assessment is also identical, or the same between them. All right. So what did we find? So this is a bunch of different papers that we've published over the years, over recent years. The first thing, just to be really clear, is that when we assess the cognition of the two groups, it's identical to each other. So we assess them around about 9 to 10 weeks post-injury, and what that's telling us is, just as we would expect from general previous literature, cognitively, the mild traumatic brain injury group is no different to the trauma control group. They've made whatever recovery in cognition that you would expect to make at a group level this 9 to 10 weeks post-injury. So they're operating as they should be in terms of cognition. What we did in this particular study was look at white matter tracts. We did tractography with, you can see them there, the anterior corona radiata, the corpus callosum, the inferior longitudinal fasciculus. We broke up the superior longitudinal fasciculus into different segment components, and we also looked at the uncinate fasciculus. What we found was that there is, across all of those tracts, that there was group differences. So there was, even 10 weeks post-injury, there were structural differences between the white matter tracts, between the groups. We found that we had reduced fractional anisotropy. I should say we were doing DTI and NODI. Reduced fractional anisotropy and increased ODI on NODI between the groups. So both on fractional anisotropy and on orientation dispersion index, there were group differences. We didn't find group differences on a number of other indices from DTI and NODI, but there were really consistent differences between the groups on across the tracts. Similarly, oh my goodness. Okay, similarly, In some work we're currently doing with a PhD student of mine, we have broken down the thalamus bilaterally into its subnuclear components. You can see that we've separated the structural volume into 8 different regions bilaterally and looked at the sub-region volumes and nodding metrics. And what we found is the same pattern, which is that we have group differences. So the red is the mild traumatic brain injury group, the blue is the trauma control group. And what we found was that we had increased grey matter volume in the mild traumatic brain injury group, not in all sub-regions, so primarily in the bilateral medial thalamus, medial thalamus bilaterally, and bilateral intralaminar regions as well. And that's just an example of the left and right intralaminar findings. And the idea, we think this might be due to the increased grey matter volume, sub-region volume, is that possibly remyelination or ongoing myelin debris is still present and being addressed. We also, this was an MPsych student, we did susceptibility weighted index, which for those of you who don't know is a way of imaging microhemorrhages, so very, very tiny hemorrhages. They occur at the junction of the grey and white matter. And what we found was that not only are you more likely to have a microhemorrhage if you've had a mild traumatic brain injury, but so you can see that on the left-hand side, that's the mild TBI group, 13 of, about 35% of the group, was with like 70 odd people, 35% of the group, total I should say, 75% of the group had microhemorrhages as opposed to 12% of the trauma control group having microhemorrhages. But much more interestingly, in my opinion, is the fact that for the trauma control group, if they had a microhemorrhage, they only had 1. For the mild traumatic brain injury group, if they had a microhemorrhage, they were much more likely to have more than 1, and in fact more than 20. So there is clearly ongoing structural difference at this point between the 2 groups as well. We then did a correlational analysis looking at the relationship between microhemorrhages and processing speed, and actually found there was a relationship there as well. So for those people who've had concussion and have microcerebral hemorrhage, they are more likely to have slower processing speed 10 weeks after the injury. So when they're at a group level, cognitively well, individually, those who have microcerebral hemorrhage are more likely to have processing speed problems. Similarly, if we look at the relationship between the structural changes and cognition from the other from other work as well, we can see a similar pattern happening, which is that when we looked at the white matter tract study, what we found was when we compared white matter tract metrics with cognitive function, consistently across processing speed, attention, memory, and executive function, the trauma control group had a completely normal relationship between white matter tract metrics and cognitive function. And consistently on all of those domains except attention, which was the problem with our attention measure, so but with processing speed, memory, and executive function, the mild traumatic brain injury group consistently had an abnormal relationship between structure and cognition. We've done this work also with a recent connectome study looking at the thalamus subregions, and don't ask me to explain the top right-hand metric. That's for people who want to know what connectome stuff is, but basically what you need to know is that edge strength is the strength of connection between 2 areas that the thalamus is connecting to. And what we found is that, as with all our studies, cognition is comparable at a group level between the 2 groups, and in fact edge strength is comparable between the 2 groups. But again, blue is normal, red is mild traumatic brain injury group. The edge strength, the higher the edge strength, the better the cognition in the trauma control group, consistently for northern and medial thalamic regions, and increased edge strength predicted poorer cognition in the concussion group. So again, it's this pattern of the relationship between the structure and the cognitive function has altered, even though at a group level, these patients have gone back to looking normal. So basically, what does that say? It tells us that neuropathology has not returned to normal by 10 weeks post-injury, although everyone expects it to have. It also tells us that suffering and concussion results in a changed relationship between structure and cognitive function, even in this period of you're meant to be better by now and you're certainly back at work and everything's completely normal. And it raises the question of whether, in fact, that return to normal cognition at a group level is happening, or suggests that it's happening despite ongoing underlying structural damage, and that perhaps that reorganising functional ability is perhaps there's some variability in that, and that maybe some patients are doing that return to cognitive normal better in the face of structural damage than other patients, and that might be contributing to why we have this variability in outcome patients. And don't break this, there
Thank you very much. Can I ask to come back or to answer some questions? Do we need the microphone for the recording? Everything will be okay. Just there's people with hearing impairments. I mean, for the question,
In terms of answering.
oh, well, yes, that's a good idea.
Thank you. My name's Eric. I'm just a community observer of all this incredible microscience stuff. Thank you very much for sharing your research. A question, sports concussions getting a lot of attention at the moment. Isn't microhemorrhaging an obvious measure of sports concussion? And how quickly could they identify it in terms of ranking the impact of the sports concussion?
So the majority of the problem with sports concussion or any kind of concussion is not the microhemorrhage, it's the diffuse axonal injury. And so that is really where the emphasis has been. Certainly with this work kind of illustrating the relevance of microhemorrhage, they may start focusing on it a little bit more. But in actual fact, with sports concussion, one of the reasons it's so different to community-based concussion is that patients typically have 20, 30, 40 concussions across their professional career. And so you have a cumulative issue going on, which is just completely different to the normal population. And that probably is happening with microcerebral hemorrhage as well, but it's certainly not the primary contributor to issues.
Oh yeah, but just as an indicator.
Well, microcerebral so the indicator of injury tends to just be happening with respect to when are they ready to go back to play, rather than neuropathology. So they don't all get scanned every time they get a concussion. It's more that functional performance is assessed, and when they're considered to be back to normal, they get to go back and play. So I'm sure it will be something that researchers look at in terms of neuropathological kind of consequences of sports concussion.
I've got a couple of questions for Kelsey. How does uncertainty minimisation relate to homeostatic set point in individuals with sensory hypersensitivity?
It does relate, yes. So the question is, how does uncertainty reduction and prediction error minimization relate to hypersensitivity?
Exactly, yeah.
It's been a long time since I've thought about this. I do have a paper on this. You'll have to come back to me later. I don't have a good answer for you right this second.
And you did have in your PowerPoint, it said autistic people act early in response to rising prediction error.
Right. Yeah.
So
they might be more sensitive to uncertainty in the environment,
Yeah.
if you relate it specifically to that, yeah.
So what's going on in autistic shutdown?
That the system's overwhelmed by the uncertainty in the environment is one interpretation of what's happening in autistic shutdown.
So there's too much prediction error, basically.
Yeah, and that the prediction error might be minimized. The perceived with the prediction error that's coming into an autistic system versus a neurotypical system might be higher, even given the same environment, based on contextual cues to the expected level of prediction error, to the ability to reduce prediction error given certain actions. So a neurotypical person might have more policies at their disposal to reduce the prediction error, whereas an autistic person might not infer that their actions have any effect on prediction error, which might lead to a higher rate of overwhelm. I think there are lots of possible explanations depending on which route you want to go.
Yeah. Can I keep asking questions, or I don't want to hog?
Thank you. I'm not a neuroscience person, so this might be very naive, but can microhemorrhaging cause CTE, or is there a link between that and...
CTE, or chronic traumatic encephalopathy, is a neurodegenerative disease. So there is a lot of lack of knowledge and a lot of over-reporting in the media around the relationship between CTE and multiple mild traumatic brain injury. So what we know is that CTE is a degenerative condition that can develop in some people who have lots and lots of concussion events. The conversion from just lots of concussions and the cumulative effect of that to an actual neurodegenerative process, which is what CTE is not clearly understood at this point. It has the neuropathological or a neuroimaging hallmark feature, or it's actually histological hallmark feature of CTE, is a particular pattern of tau deposition at the in particular areas of the cortex, not microcerebral hemorrhage. So microcerebral hemorrhage is distinct from and separate to CTE, but it certainly could be contributing to the cumulative effect of individual concussions in people who have multiple concussions.
Hi, Kelsey. Great talk. A question, have you looked into sort of avoidance symptoms? Because one way to resolve prediction error is never to get exposed to prediction error. Could that be a mechanism that kind of moderates your differential relationship between self and different diagnoses?
Yeah, I think it's an interesting question. I think it relates to criticisms of predictive processing of the dark room problem, which is why don't we all just go sit in a dark room and where we'd never get prediction error. I think the usual response to that is that we don't expect a lack of prediction error. Like we deal with complex worlds. We are evolutionarily designed to deal with complex worlds, and that involves some level of prediction error that we can control some of and not other bits of it. But I think it would be really interesting, because that is one strategy, is to avoid a complex scenario or to choose environments that are more simple and have more predictable kind of causal relationships between the elements of the environment. So I do think it's an interesting path to follow up on in the future as another option for minimizing prediction error related to the self. Yeah, interesting. No, I haven't done it.
This question is for Robin. Can you speak to whether or not the individualization of the stimulation site has any association with the longevity of results that you're seeing? And could you posit possibly why people with more complex comorbidities are having fewer recovery?
Thanks. Two really interesting questions. I guess our research is kind of still at the early stages, so in the early kind of years. We don't yet have data around the longitudinal or longevity of effects with personalised versus non-personalised brain stimulation. We will collect 6 months of follow-up in our clinical trial, initially it was meant to be 12 months, but yeah, we didn't get enough funding to do that. And then in terms of comorbidities, I think, currently, I mean, our approach is targeted towards the subgenual cingulate cortex. And so I think in that case, we can target symptoms relating to negative affect, or negative emotions, but it's not tailored to other conditions or comorbidities. So I think in that, having said that, the confusing part is why does it still not work as well for negative affect in those individuals? And yeah, we don't really know. I think maybe there's kind of too much going on, or maybe they need more intensive, a more intensive course of treatment, or maybe we need to address other issues that we're dealing or other comorbidities. But yeah, that's definitely something that we're looking at. It's just really complicated, unfortunately. Yeah.
I also have a question for Robin. Yeah, kind of a follow-up to that one. So you're focusing on the subgenual cingulate.
Yeah.
So one interesting thing is that some of the deep brain stimulation clinical trials for the subgenual cingulate have basically shown no effect. And that's sometimes been explained as a kind of a failure of personalisation, like we didn't put it in the right spot in the subgenual cingulate for this person. I wanted to ask, based on your research, first, is that sort of, would that be an endorsement you'd accept? And second of all, maybe this is slightly more pointed, like in general, how can we as scientists know the difference between a treatment that has failed versus a treatment that is not sufficiently personalised?
Yeah, okay, two good questions. So it's true, with some of the, so the initial, or the first randomised clinical trial, multi-site clinical trial looking at the effect of deep brain stimulation targeted at the SGC. Yeah, it did fail, but then there have been follow-up studies showing actual response, like a delayed response in those individuals. So actually, it may not have been a failed study. And the other thing is that now, with deep brain stimulation, they are starting to, so they previously they were essentially targeting like a single site with DBS. Now it's becoming more tailored as well, and that actually seems to be a lot more effective as well. The second question, sorry, what was it again?
It's just how can we know when we've chosen the wrong site versus when we just haven't tailored it enough?
I think, I mean, once we've done this clinical trial, we'll probably have a better answer for that one. I think, yeah. Ongoing research.
Question for Jay. So the results you were looking at, they were all at 10 weeks. And I was wondering whether you think some of it will extend beyond that? Will it be long-term, or will there be plasticity eventually that will resolve?
Well, if anyone's got some spare money, we can help answer that question. We are collecting data at the 12-month follow-up period. It's very slow going, getting people to actually come back in and contribute to, 3 hours of testing and a 45-minute scan that far down the track, but we are slowly accruing data. So things like the microcerebral hemorrhage, that's permanent. Like there's no coming back from that. That's a permanent change. In terms of some of the thalamic volume stuff, that might resolve because it is sort of potentially about ongoing myelin readaptation or whatever. In terms of the microstructure changes, I don't know, but my guess is that probably some of this stuff is permanent, to be honest, because we're quite a long way down the track. We're at the end of what you would consider to be the rapid recovery in terms of neurological recovery period. So, neurological recovery will slow from 3 months to 6 months and slows a lot more again up to 12 weeks and then you're done basically. So yes, there is more room for neurological recovery to happen, but the big amount of it is done by 3 months. So my guess is that probably some of these structural changes are ongoing, but it's a guess.
I've got a follow-up question, but you found that they had a different association between cognition and white matter structure. Do you think it's because that reflects reorganization of function? That reflects reorganization of function, or is it just the measures are a bit noisy because of the trauma, and so the association is just a bit like it's not coming up because of the measure being a bit different?
No, I suspect it's probably functional reorganisation that's going on. Whether it's structural reorganisation, I don't know, but I'm guessing that it's functional reorganisation is a big part of what's happening, and that therefore the way that they're going about their cognitive tasks has changed to compensate for what has happened. Yeah, that's my guess.
I've got questions for Rob. With TMS personalisation, is the inter-individual variability segmented into clearly defined phenotypes, or is every individual completely unique from every other?
Good, very nice question. Yeah, well, if we're looking at just overall reduction, so our main clinical correlate is the reduction in overall symptom severity. And in that case, yeah, there's no real phenotypes. It's just kind of like a scattering of different targets or different optimal stimulation target sites. Yeah, beyond that, maybe we can look at at biotypes and subtypes and that kind of thing. Again, that's something that with the data set that we're collecting, hopefully we can do a bit more work on that. But yeah, nice question.
Okay, great. Well, thank you very much. Please join me in thanking the speakers. And now we have the morning tea break. So please come back here for 11:30, and the tea is downstairs in the Hub Cafe.
This punctual group. Thank you all for coming back on time. We have a really exciting second session for you on cognition. We're going to have 2 speakers, and we're going to hold questions till the end. We're going to invite Robin and Kelsey back up to do a panel at the end of these 2 talks. So if you had questions that weren't answered earlier, you'll have another opportunity. Our first speaker today is Dr. Dragan Rangelov from Swinburne Institute of Technology. He's a senior lecturer in psychology and cognitive neuroscience, and he earned his PhD in systemic neuroscience from Ludwig Maximilians University in Munich, and he has a background in neurocognitive psychology. His primary research interests lie in the neural and cognitive mechanisms of perception, attention, memory, decision-making, and metacognition. So there's something here for everyone. He employs behavioral paradigms, brain imaging, including EEG, MEG, and MRI, and non-invasive modulation techniques, including TMS and TCS and neurofeedback. And he uses these techniques to probe how the brain adapts to environmental change and supports flexible cognition. He leads the Economic Brain Lab at Swinburne, focusing on how risky choices are modulated by higher order cognition, things like metacognition. So we're really excited to hear from him today. Please join me in welcoming Dr. Rangelov.
Wow, that was a mouthful. Good. Before I start, I just want to thank the organizer for the kind invitation. It's lovely to be here. I relocated to Melbourne just recently, so most of the work, all of the work I will be presenting today was done in a collaboration with Jason Mattingly at the Queensland Brain Institute. And the main topic of this research, or this kind of line of research, is metacognition, or more specifically, neural mechanisms that inform or predict changes of mind. So before I dive in, I would just like to use this problem as an illustration. I find it kind of instructive in terms of what changes of mind are. So I could imagine most of you have heard of the Monty Hall problem, but let me guide you through that. So this was actually a sort of real TV show in which the host would offer very attractive prizes to the contestants. And the task was that they needed to choose at random one of these 3 doors. Behind one of those 3 doors was a very attractive prize. So let's say the contestant would choose this blue door, and here the real game starts. So after the initial choice, the host, Monty Hall, would have opened one of those doors and revealed that there was no prize behind that door. And then the contestants were given a choice again, whether they wanted to stick with their original choice or to change their original choice. So what would you have done? Can I see a show of hands of people who would like to stick with their choice? There's at least 1, 2, 3. Good. A few. And who would like to change their mind? Okay. So I guess this intuition, and potentially you have read about it in terms of statistical regularities, is that people potentially should change their mind. And there's also statistical reasons for that, which we can discuss later on. However, research on changes of mind did not really use Monty Hall problem, but there has been a lot of research on that, and that research we reviewed a couple of years back has shown that changes of mind as such are a special type of choices, of decisions, that alter the initial choice following some process of metacognitive deliberation. So people do extra work in order to decide whether or not to change their mind. And what research has shown, which I find interesting, is that these decisions are infrequent. People are allowed to change their mind. They are typically corrective, so people tend to improve their performance, and changes of mind obviously are pervasive. So from changing your mind about the menu item you're ordering to changing your mind whether to swipe left or right, they are all over in our daily lives. So one question that you can ask is, what is the cognitive mechanism by which we sort of make a choice to change our mind? This is obviously a bit difficult to investigate because changes of mind are infrequent, so these behaviors are somewhat difficult to elicit. In this particular study, which is maybe 15 years old, the authors actually developed a very nice paradigm in which participants were supposed to move the pointer, which is here the red circle, in the direction of these 2 yellow targets, depending on the motion direction presented in the middle. And what the typical situation, or what investigation of the motion trajectories as people were responding has shown, is that here there were 2 groups of trials. Trials in which people started the motion and continued moving in the desired direction, but there were also many trials in which people kind of initiated motion direction toward one target, and then they changed their mind mid-flight and ended up in a different target location. And this paradigm was very useful because such changes of mind detected by changes in trajectories were much more frequent than if you just ask your participants, would you like to change your mind or not? So how can we explain the temporal dynamics of how people decide to change mind? One can potentially apply such computational modeling of behavior, which I'm sure many people in the audience are aware of. And one such model is so-called drift diffusion model, which essentially postulates that decisions unfold in time. They take certain time during which some evidence is accumulated towards some of the decision boundaries for one or the other response. The main idea here, in terms of computational mechanisms of mind changing, is that the same mechanism is engaged again after the original response, but in this kind of scenario, which is indicated in the pink area, people actually accumulate evidence whether or not they should change mind. And you can see here in this temporal trajectory of so-called decision variable that for those trials in which people change mind, this is the pink or violet one, that trajectory changes direction. If you record brain activity and try to sort of identify neural correlates of such decision variable in brain, there have been many such neural correlates reported. So we are not really short in such things, but we observe such changes in these neural correlates of decision variable both before the response, shown here on the left, and after the response. In terms of neural networks involved in changes of mind, people have shown that it's about a relatively widespread network, primarily situated in the frontal brain areas. So we have learned a lot about how people change minds. Now, when we started this whole line of research, we were actually interested to potentially inform the field in terms of characterizing some precursors, some predictors of changes of mind. And one of such predictors could be potentially decision uncertainty. So when you are uncertain about your decision, you should be more likely to change your mind. However, this almost sounds like a tautology. It's kind of course, that if you are uncertain about your decision, you would be more likely to change your mind. So how can we sort of escape that tautology? So modern theories of decision-making, and more broadly of cognition, as we heard earlier today from Kelsey, postulate that the human cognitive system represents sensory input in a probabilistic way. So you can think, for example, of a task in which you show some motion direction, and the cognitive system starts to accumulate that iteratively over different samples. So the main idea is the representation, the cognitive representation of such motion direction will start initially as being very wide and potentially far off the presented motion direction. And we can characterize two parameters of that cognitive representation. One would be obviously the peak or the location, and another would be the width of that representation, which in this case we can use as a good measure of decision uncertainty. As we accumulate our evidence, that representation, cognitive representation, should sort of wobble around the motion space, approaching the true accurate motion direction, and also decrease in width. So it will become more precise. And indeed, you can model such drift diffusion process by assuming that the cognitive system is actually accumulating or reducing uncertainty of these representations, which we have also shown relatively recently. Now, in the first study that I'm going to talk about, we ask a question, do decision makers use uncertainty to inform changes of mind? So that was the main question here. And we developed this paradigm, which will be the same across studies. So let's spend some time just discussing it. So we used motion discrimination task, in which we presented this field of dots that were kind of moving randomly initially, together with this sort of colored grating in the middle, which I will explain shortly. After a little while, we started by presenting coherent motion signals in the dots, and participants were asked to judge whether the motion the motion signals of those dots were going more towards the orange portion of this colored grating or towards the blue portion of the colored grating. This is kind of a methodological trick because you can then nicely separate response preparation and motion signals. It's just there for the sake of methodological ability to identify stimulus-related and response-related processes. We asked participants to try to make speeded responses after they have responded. We actually stopped all those dots, and we presented them with a question, as illustrated in this scale in the final display containing the yellow circle. People had to report the degree to which they would like to change their mind. And going from left to right, that degree went from 0 to 1. We hope that this particular way of measuring change of mind would help us identify more frequently trials in which participants actually wanted to change their mind rather than asking them for yes or no responses. We manipulated two aspects of task difficulty. So one is motion coherence, or the percentage of dots that are moving in one direction. And another, which I think is very interesting, is the offset between the direction of motion and the orientation of the response criterion. This kind of doesn't impact really any sort of sensory processing, but it does produce measurable effects in terms of response times. And you can maybe think of it as a more sort of a manipulation of decision difficulty, if you wish. We recorded the EEG as participants performed the task. And just so the conventions of in terms of data presentation would be the same. So here I'm showing on the left is the percentage of accuracy. And we see that there was a very strong effect of manipulation of task difficulty, both in terms of the offset and in terms of coherence, which may be a bit more difficult to see. And interestingly, the effects of upset were larger than manipulations of coherence. We also observed slowing down in reaction times of correct responses as a function of an increase in task difficulty. Now, the main here parameter is change of mind responses. And here I'm showing on the left figure a distribution of such change of mind responses across all trials for 2 participants. And you can tell that there was a significant individual variability. Some participants used the whole scale in a kind of scale-like manner. And some participants used just most frequently 3 different kind of categorical responses. If we sort of Z-score all these responses in order to remove individual sort of biases in terms of overall readiness to change mind, as plotted here on the right, we do see that there's a difference in change of mind scores between correct and error responses, which is a good thing because it indicates that people actually are able to respond in an adaptive way, in such a way that they are less likely to report strong desire to change their mind following correct responses. Now here you can also compute contrast between different conditions, and what we observe again is that change of mind responses as well are impacted by task difficulty. And in addition to that, you can compute some measure of metacognitive sensitivity, if you wish, which is a change of mind difference score, or the difference between change of mind responses following correct and error responses. And we see here that these parameters are all above 0, telling us, again, the same thing. Yes, people are able to decide to change their minds when that's beneficial for them. Now, the main idea here is, let's see what's happening with the brain data. As I mentioned, we were using EEG to record concurrent brain activity. Here I'm plotting these topomaps. In the upper row, there's a stimulus onset lock topomap, so when the coherent motion starts. And in the bottom row, there's a response onset topomap, so time locked to the time when people actually press the button. We see here interesting parallels, specifically this kind of big blob of positivity around centroparietal regions, which has been well described in the literature under different guises and different names. People call this centroparietal positivity if it happens in a stimulus-locked way, or maybe error-related positivity if it happens after the response. What we had done in terms of analyses, I'm not going to plot evoked responses. Rather, we were interested to see whether measured neural activity across this cluster of centroparietal electrodes is predictive, if at all, for subsequent change of mind responses. So what I'm plotting here are regression rates telling us how much neural activity measured in this stimulus-locked time period predicts people's desire to change mind. And what we see here is that, yes, there is a significant relationship predominantly contained to correct responses, and that relationship is negative, as seen here in the magenta line. The similar pattern, but much more variable for error responses as well. If you look at the activity that was time locked to the response, so everything from the vertical dotted line here towards further in time happens after people have already responded. Here we see a different inversion in this pattern, in that what we see here is strongest relationship between brain activity and subsequent changes of mind is being restricted to error responses. So there's this kind of nice sort of dissociation between neural activity predictive for changes of mind following correct and error responses to kind of pre and post stimulus time periods. Good. So in the second study, we were interested in characterizing which conditions, if at all, promote sensitivity to uncertainty, to decision uncertainty. And to do that, we actually went on to manipulate speed accuracy trade-offs, because literature shows that there may be differences in metacognitive sensitivity depending on whether people are pressed for time when they are responding. So in this study, we use the same paradigm. However, here we introduced sort of monetary rewards depending on whether or not people correctly wanted to change their mind. And we also introduced two different conditions in which we had long and short response deadlines. together with the sort of EEG recording. So here I'm just plotting behavioral results. We replicated most of the results from the first study. The main kind of take-home message to show you from this set of results is that our manipulation of speed accuracy trade-off was effective. So people in the short deadline indeed were faster and less accurate than people in the long deadline condition. Now, in terms of changes of mind, what we observe here, if you just focus on the bottom row, this is the change of mind difference score. We actually observed that people in the short deadline were more metacognitively sensitive than people in the long deadline condition, which was actually reported in the literature. So what's happening in terms of EEG data? What we show here is, again, stimulus-locked regression with subsequent changes of mind from the same kind of set of posterior electrodes. And again, we see that these stimulus-locked time sort of traces are predominantly predictive for changes of mind in correct response trials, whereby the post-response time periods are predominantly sensitive or predictive of changes of mind following incorrect or error trials. And interestingly, we also observed this interaction between different speed pressure conditions, but selectively in the post-decisional time period. Good. I don't have a lot of time to guide you through this. I'm running out of time, but in the last study, we essentially tried to see whether we can improve people's metacognitive sensitivity using cognitive training. We developed a training paradigm that used as a kind of outcome measure performance in the task I already described. And here we had somewhat contrived and complicated training paradigm in which we essentially had two groups, which we potentially, unfortunately, decided to name experimental and control group. Both of these groups had to do some task. The main difference between them is that in experimental group, people had an opportunity to choose freely which task they wanted to do, and presumably that was supposed to train them to kind of decide on the basis of their awareness of decision uncertainty to potentially pick the one that is less uncertain. So that was the main kind of point of the manipulation. In terms of behavioral findings, and here is where the things sort of get hairy, we observed comparable training effects for both these groups. This was kind of good. There were training effects. People did improve in metacognitive sensitivity. However, there was no interaction with the group. So no matter what sort of training you give people, they get better, really. So that's kind of heartening, but it didn't really sort of highlight the differences in groups. What we have done next, we have used sequential sampling evidence accumulation modeling, trying to untangle potential sort of parameters that drive these learning effects. This model fitting was behavioral. We got good model fits. And what I'm showing here are sort of distributions of the drift rate parameter across different conditions. And without going into much details, the most interesting thing is this difference here, in which we actually see that people in the nominally named experimental group improved in their drift rates following training, in contrast to people in the control group, which didn't improve as much. So it seems that there was a group-specific training improvement that actually targeted drift rates. However, we were not able to see that in the behavior for other reasons. Finally, if we check the evoked responses, here again, these are regression weights. In the top row, this is just pre-test. We do not really see strong differences between groups, which is kind of okay. After the training, what we see, that's in the bottom row, we see these very strong differences between groups. And these differences kind of speak to the fact that although people overall improved, the mechanism of that improvement was different. So that's the main idea. So when people are given free choice, the mechanism of the improvement in their metacognitive cognition was different from when people were not given a free choice in the training session. Good. So in terms of general summary, so what have we found here? We did find that changes of mind rely on decisional uncertainty, which is heartening. That sensitivity was it was possible for us to improve that sensitivity with training, which is also good because that may have implications for training in some risky professions such as, say, air traffic controllers. And in terms of open questions, one may ask why, if we know the changes of mind are so beneficial, why are they so infrequent? There could be a host of different reasons for that. And one could also ask a question whether there could be special groups along the lines of what Kelsey was saying this morning that potentially could be differentially sensitive to training and improve differentially. With that, I would like to thank you all for your attention. I'd like to thank our sponsors and the team. And in particular, I would like to highlight Caleb, who was like the best PhD student you can imagine. And he's now doing his postdoc at UNSW. Thank you.
Thank you so much. I have so many questions about why I change my mind so frequently. Okay, as we switch over, our next speaker is one of our very own, Associate Professor Natalia Igorova Brumley. And I have had the pleasure of introducing her a lot lately, so she challenged me to memorize her bio, which I have not done. She completed her PhD in Cognitive Neuroscience at the University of Cambridge, and she received postdoctoral training in pain neuroimaging at Massachusetts General Hospital and Harvard Medical School. After she's moved to Australia, she worked at the Florey Institute of Neuroscience and Mental Health before starting the Pain and Cognition Lab at the University of Melbourne. She combines cognitive and clinical neuroimaging to understand how pain impacts the brain and how cognition alters pain processing. She's currently a Dame Kate Campbell Fellow and an Australian Research Council Future Fellow, and she investigates the neurobiological mechanisms of the interaction between pain and sleep, which she's going to talk to us today about Bayesian inference in judging the pain of others. So thank you. Please welcome Associate Professor Brumley.
Sorry for the introduction. Can you hear me? No, not really. This one. Yeah. So let me just...
I think that one seems to be a dud, and this one's on.
Is it? Yeah.
We heard you taking it off.
Sorry, I tried to have her pre-prepare, but that does.
Okay. Let's try. Sorry. Yeah. Can you hear me now? Yes, perfect. Thank you very much. Okay, so you were probably wondering what is a pain talk doing here, but I run pain and cognition neuroimaging lab where we're trying to understand how cognition actually affects pain. So this is going to be a talk outside of my depth because it's on Bayesian inference and I'm not an expert in it. However, in the hub, we have so much expertise in this area, and I will deflect all difficult questions to my colleagues. So with that in mind, I also want to preface it with the fact that this is not a complete body of multiple studies, but just a couple of new studies that we ran, and it's still unpublished work, so don't judge us too harshly, but I'll welcome any feedback. So when I talk about Bayesian inference, thanks, Dragan, for doing the introduction. But what I will mean is that typically we have the perception combining both our expectation and some sensory input that we receive from the environment. And Bayesian accounts have explained a lot of different phenomena in behaviors, but what we are interested in is how it's been applied to pain. And there has been quite a bit of work from about 2010 up to now, and it's ramping up gradually to try and explain pain with respect to how your sensory input might be different to your expectations and how it could create different perceptions of pain. And for example, here I'm showing you a study by Christian Büchel in 2014. And I don't know if you can see it, But normally you would have no expectation of pain, and then suddenly you are given a placebo or a drug, and you don't know whether it is a placebo or a drug, but you already form some expectation of feeling better, and that shifts your perception possibly towards less pain. And then finally, if you are told that you are given a powerful drug, you are also very likely to feel less pain just because your expectation is now a lot sharper. So in this, I didn't see if it's working. So I'll just try to find my mouse. Yeah. Okay. No, it's not very. Okay, so in here, the sharpening of the expectation does affect your pain perception in a stronger way, shifting the posterior even further away from the original and towards the less pain. So as I mentioned, most of these studies that have been run so far have demonstrated that shift towards the prior when we're talking about pain. A lot of the Bayesian inference was, again, demonstrated with as the shift towards the prior. However, most of these studies involved experimental pain. So what I mean by that is that they explicitly changed your perception of pain. They implemented paradigms which delivered pain stimuli to participants, and they actively change the perception that you had. So that is one important thing to keep in mind. The other point to keep in mind is that although all of these Bayesian inference and pain accounts were postulated as very relevant to chronic pain, most of these studies, if not all, have actually been conducted in healthy participants. So they have not really been tested with real pain in real people who experience pain, so in chronic pain. So this is important because chronic pain is not the same as experimental pain. And here I'm just showing you, for example, the time course of a stimulus and the response to experimentally induced pain in chronic pain patients and in normal controls. And in fact, it is pretty much the same in the top graph there. And here in B, spontaneous pain, which is the endogenous pain that's experienced by a chronic pain person, is very different to the thermal pain that is induced in our experiments. And often chronic pain patients and controls might have a completely normal experimental pain response, but it's really the chronic pain component is not so much about the sensory input. What I also wanted to show you is this graph that shows how brain reorganizes in persistent pain. At the top there, you have persistent pain patients, so who had pain at visit 1, and at the bottom you have recovered pain patients. So from visit 1, they were scanned. If you look at visit 4, so in those recovered, there is no more pain. But if we look at which networks the pain is persistent, sorry. Oh. Going back, it's actually the green and red, which is emotion and reward systems rather than the pain sensory systems where pain persists. So we really need to appreciate that chronic pain is a lot more about this cognitive emotional appraisal of pain rather than actual sensory inputs. So, and finally, one last slide to really convince you. This is a diagram of brain regions that are involved in chronic pain transition. And the risk is really in these green and blue areas rather than the red ones being typically associated with the intensity of pain, with the location of pain. But it's really in this reward and appreciation of pain where it changes. So now that you're convinced that pain is different, experimental pain, what do we know about ways to actually study Bayesian inference in chronic pain patients? So this is a set of graphs that show you a paradigm that has nothing to do with pain, but was developed as a paradigm to study Bayesian inference in perception. And here it was developed by Villaris, but Marta Garrido's group have implemented it in autism and in schizophrenia psychotic features. And I'll just start by explaining it a little bit in order to show you what we have done with it. So in this paradigm, you see a 2x2 design where the likelihood and priors can be manipulated. The task goes like this. You are showing some splashes. So the task is of a thrower throwing a coin, and what you are given is the distribution of the splashes of that coin when it falls. So and the task is to guess the location of the coin that landed in the water and produced the splashes that you are seeing. And what's also manipulated is the accuracy of the thrower. So you get the really precise thrower or somebody who's not very precise. And then what you can calculate for each condition is the departure from perfectly following the priors. So that would be the complete horizontal line. If you only relied on the prior, you would have it completely flat. If you only relied on the sensory information, so that the distribution of splashes, you would have a completely diagonal line. But what is found is that you occur somewhere in the middle there, and the slope is really showing us how much you rely on the likelihood or prior information for any particular condition and any particular individual. So what has been done with this previously is the for example, correlation with psychotic features, where the more you rely on the sensory information, the more psychotic features you have. And those findings mostly come from the conditions with the wide sensory information. So now this is about psychotic features, about autism, about perception. How can we use it for pain? Well, some of the work that I've done early in my postdocs was actually designing several paradigms where you wouldn't need to induce pain in somebody to study pain, but rather show people videos of people experiencing pain. And what we've previously done was to compare observational learning of pain to the direct experience of pain. And other groups have also demonstrated that you don't even need the information about the participants' facial expressions of pain in order to learn about their pain. You could, for example, show the ratings of somebody to some unknown painful stimulus, and participants are able to learn about the degree somebody is in pain from just looking at the distribution of their pain ratings. So we use this information that you can learn about pain without experiencing pain, and implemented the new design of the coin task, this time completely basing it on the pain paradigm. So I'll go over it. Instead of having the thrower and the coin, we have a person who's experiencing pain and then showing their pain ratings to a particular stimulus. All of these ratings and all of these models are completely fake. So this is completely generated data based on distributions that we want to be preserved. And we show them so that the likelihood information, which is the spread of the pain ratings, is either wide or narrow, and the accuracy with which people rate their pain as precise or imprecise. So again, we manipulate, just like in the previous design, we manipulate both the prior and likelihood. Again, we had to do a little bit more teaching as to what pain ratings are, and we took the liberty of teaching people that it's a Gaussian distribution. It's not quite, but for the purposes of this experiment, it was assumed that on average people would rate pain as something painful at about 41 degrees. So the task was to guess the temperature based on the distribution of pain ratings and the rater's accuracy. And we completely copied the paradigm and the organization of the trials from this previous set of studies that Marta's group has conducted, having block design with the narrow prior block, wide prior block, and for the different participants rating pain. So now in terms of the sample that we had, we wanted to actually look at chronic pain and explore whether there is the experience of chronic pain influences these inferential strategies. And we had access to 152 participants who have completed the study, and 57% of them were females aged between 20 and 87 with a mean of about 57. And you can see where most participants expressed pain, so a lot of low back pain, a lot of knee pain, and some headache. So we characterized their pain using the brief pain inventory, which is able to differentiate between pain interference and pain severity. And we computed the sensory weights for each condition for each participant. So again, sensory weight, those were the slopes that I was talking about, and roughly they provide information about how much a participant relies on the sensory information versus the priors. So with respect to the sensory weight distribution per condition, they were not quite optimal, but they were consistent with previous studies, so similar results. So this was a pre-registered study. We did expect people to have a prior shift because, again, this was consistent with some of the previous studies in healthy participants. However, we had our main analysis being the total pain in the brief pain inventory, and it was indeed a negative association as we expected. But what was interesting for us was whether the pain severity, so the sensory information or the interference, was predicting these results, and people were more likely to rely on the priors if they in relation to pain interference rather than severity. So we did not find any association with the severity, but we did find it with the interference. So now, in terms of the, typically we can differentiate between whether the shift towards the priors is explained better by overly precise priors or by noisy sensations. And in a lot of the previous studies, there would be one explanation for this, and that is overly precise priors. And that is because priors were actively shifted and the sensory information was presented in a very strong way. So we wanted to know, and again, if possible, whether in the chronic pain condition where people are not experiencing any induced pain, whether that would also be the case. So when we looked at our results by condition, and I'm showing you the interference plots, it's really in the wide likelihood conditions that we saw the results. So there was more of an association. And when we tried to compute prior variance and related to pain, unfortunately, there was no association between prior variance and the pain interference, as we would expect. So without having the certainty, without having conditions that explored subjective likelihood, it is hard to conclude with certainty what the situation is, but it's definitely showing that it might be not only the overly precise priors, but also possibly the noisy sensations that are driving these effects in chronic pain. So the interim conclusion would be that the participants who experience more pain rely more on priors than sensory information. And pain interference shows a stronger association with the shift towards priors. If you remember my plots about the sensory versus the cognitive, evaluative, and affective networks that are involved, it is very likely that it is this shift towards the interpretation and experience of pain interfering with your daily life that's really driving this effect. And then the association was stronger in the wide likelihood conditions with no association with prior variance, possibly favoring the noisy sensation hypothesis, but again, it needs to be further determined. Now I just wanted to share with you a little bit of data on the healthy participants. This data set we had even prior to this pre-registered study as we were optimizing the paradigm, and also wanted to see what the association between pain and the the sensory weights were in the normal population, and that is the university student population. We had access to 603 participants, mostly female, aged about 20. And of course, in this population, we don't ask people about the severity of their pain. Instead, we ran it with a somatic symptom scale, which just asks how much or how frequently have you had in the past 7 days things like stomach pain, back pain, headache. So it probes a little bit some information about pain without actually requiring you to have chronic pain for several years prior. So we had this questionnaire, that was given to both cohorts. So I'm just going to present some results. Again, this is the distribution of sensory weights across the 4 conditions for each of the groups. The chronic pain you just seen, healthy participants behaved very similarly, and there are a lot more dots, as you can imagine, with 600. So now what we could do, we couldn't really do a proper correlation of symptoms because there would only be a little bit of pain in this large student population sample. But what we could do is, and the distribution of those numbers would be insufficient to really provide for the variability. So instead, we just grouped them as low pain and high pain. And again, in both groups, we observe differences in all conditions pretty much between low and high pain groups in their dependence on sensory weight, with sensory weight reliance going down in the higher pain group. But you can see that the effects are a lot larger in the pain group, and they are very small in the healthy participants. And because we had such a big sample, we do have statistical significance, but the effects are tiny. So what we also could think is that it could be related to performance accuracy, kind of differences. And without going too much into this analysis, which is not complete yet, the accuracy was high across both groups, and it was really different across the different conditions, but really high overall. So in conclusion, we replicate this previous finding of a shift towards prior for pain. But this time we are doing it using a completely observational paradigm. We do not present any painful stimulation to these participants. So this shift towards the priors can be observed even with no physical pain. And an important conclusion is that people who have pain also project their inferential strategies onto the pain of others, because they were asked to judge the pain of others. So it's not only their own pain, but potentially the pain of others that might be subjected to some changes in how it is perceived. Finally, we looked at it in a chronic pain population and saw that it correlates more with pain interference, and also potentially this shift toward the priors is likely resulting from the flatter likelihood in this population. So this is something that needs to be further investigated, and I'll leave you with the limitations and future directions rather than firm conclusions from the study. So we don't have the subjective likelihood condition where it would be something without the prior manipulation to compare subjective priors with, so something to do. You did not see any brains from me, unfortunately, today, other than from previous studies, so perhaps neuroimaging to follow. And then we designed a completely new task, although it is mimicking the perceptual task, the throw a coin task, but we are reporting different results, but is it the outcome of the changed task, or is it truly something that's pain-related and it's in the particular direction? So, but perhaps comparing them within one population would be helpful. Finally, we ran it as an online task, and it was great because we could reach people, but the completion rates were really low because it is a very challenging task for 57-year-olds on average. So this is something potentially simplifications of the paradigms will be useful. So with that, I'd like to thank my Pain and Cognition Neuroimaging Lab, and I see a few members in the audience. Thanks for coming, guys. And especially Adrian Gonzalez and Claire Lynn, Alina, Loza Konovalov, and Ali Gu. I also want to thank my collaborators who are really the brains behind the Bayesian stuff. Thanks, Marta and Sophie particularly, and the funders, the Australian Research Council in particular. Thank you.
Amazing, really interesting talks. If I can ask Robin and Kelsey to also come back up and we'll do a panel. Maybe, these chairs seem very tall and crowded, so you guys can stand or sit in them, whatever you'd prefer. Okay, and we'll go ahead and start with questions. And although I appreciate the eagerness of questions, we'll do one per person, and then if we have time, we'll go back around.
Thanks. Question for Dragan, really interesting talk. The question is about the theoretical mechanisms that you see as underlying change of mind. So you were relating change of mind to uncertainty and uncertainty to evidence accumulation in a diffusion model. And that's the sort of the problematic thing, as you're undoubtedly aware. Uncertainty is clearly closely related to confidence, and there has always been a problem of how you think about confidence in the classical two-boundary evidence accumulation model, because the classical interpretation of a log likelihood accumulator is that the response is always made with a constant likelihood at the time that hits the barrier. So the confidence and or the uncertainty should be the same. So the question is how you're thinking about what's the mechanism that's giving you the change of mind. There are multiple ways out of that. I can think about 3 or 4, but I'd be interested in the way you're thinking about it.
Excellent question. Obviously, So if you always need to reach certain threshold, then where does this variability come from? I don't necessarily have a good answer to that question. I can maybe speculate. I think there's a lot of insight to be gained if you kind of correlate things across trials, because yes, hypothetically you need to reach the threshold, but we do see large variability in trial dynamics. So we do see large variability in response times. Presumably those drift rates were on those trials variable in some sense. And in that sense, our effects in terms of training results did indicate that it was the drift rates that were sort of shifted for our nominally called experimental group. So I think, yes, we can run in circles in terms of if you pose a problem in the sense of a theoretical model predicts fixed threshold, whereby we are not necessarily seeing indication of that fixed threshold in the behavioral data. I don't have a good answer to that, but the main idea would be maybe looking into intertrial variability, at least I find, would be promising to address that question. So that's the best I can offer you right now.
As I said, there are probably about 3 or 4 ways out of that question, out of that problem. There's the Pleskac, Boosmeyer, that it's all post-decisional evidence accumulation. Cross-trial variability and drift rate also potentially gives you a kind of proxy for confidence if you're thinking the drift rates vary across trials.
All good points. All good points. Mostly people that focus on post-decisional evidence accumulation in favor of changes of mind or not postulate again yet another fixed decision threshold. So it's in some regards that's kind of kicking the can down the road.
Okay, thank you.
Yep, I've got a question for Natalie. Isn't it a bit of a catch-22 because your priors are your previous sensations of being in chronic pain? So because you were previously in chronic pain, you have your Bayesian inference that you're going to continue being in chronic pain because those are the presumptions of your system, basically. And how would you get out of that, basically?
Thank you very much. This is a good question because that was the purpose of the study, to actually demonstrate that that is the case for this population. Again, previous studies have been run in healthy participants postulating that it's the right theoretical framework, but there have not been any experimental validation of that. There hasn't been anything that's been shown in chronic pain. So this is the first step to demonstrate that indeed there is a shift to priors in this particular task, but potentially you could see it across different tasks should you run further experiments. What we can do with this now is to potentially think, what is it useful for? So for perhaps predicting how much pain a person is experiencing, whether they are going into the direction of persistent pain versus recovered. So this is one option. The other is just to explore the theoretical framework of Bayesian inference in pain, but in a chronic pain population, and try to understand perhaps some individual differences with it. This is something stable that we can look at whether this could be an outcome of a clinical trial looking at changes in some Bayesian inferencing that's intended to improve pain. So basically, this is the foundation to demonstrate this indeed in chronic pain population, and then try to see whether this could be an output that we can use in order to outcome that we can use to predict pain or predict transition to chronic pain or recovery for that matter.
And I'm sorry, I didn't know that Associate Professor Jay Anderson was still going to be here. So thank you, Jay.
Hi. A question for Natalia. Just following up. I'm wondering if you have any sense for whether people's with chronic pains priors are sensory modality specific. So nociceptors versus proprioceptors is kind of where I'm going.
This is an excellent question. And again, this is slightly related to one of the limitations that I mentioned, that we came up with a pain task to probe these. But if we were to give somebody, for example, a regular perceptual task about the temperatures or about their feeling something interoceptive but not pain related, would they still have shown the same shifts to the prior? So whether it's pain specific or not is something that we haven't yet explored. My guess would be that it probably would be something that's more specific to the specific symptoms that they're having, which is pain, and not something to do with, I don't know, the weather. But equally, this is an empirical question for which this paradigm would work should we run it across the different modalities.
Hi, question for Dragan, really interesting talk. You mentioned something about short guidelines and long response guidelines. And I was curious, you said in the short guidelines, there's more metacognitive sensitivity in the participants? Yes. Yeah, so I was just wondering about that, and yeah, just wonder.
Yeah, that's a very good point. So the literature, as well as our findings, seem to indicate that people exhibit higher metacognitive sensitivity when they are pressed for time. So that's kind of consensus in the literature. We were not the first ones to kind of report that. Why that exactly is the case? There could be different reasons. There's starting from relatively kind of trivial reason how metacognitive sensitivity is estimated, and it has to do with the likelihood of errors and some, I can go into details there. What I think our results show in an interesting way is that going beyond that behavioral kind of explanation, we also show differences, and specifically in interaction between response deadlines and the sort of difference between neural time traces for correct and error responses. So in that sense, there is, I think, going on top of that, additional neural generator that may improve cognitive metacognitive sensitivity when you're pressed for time, which is not trivially explained just by those kind of statistical regularities.
Thank you.
Thanks. Yeah, question for Dragana. First of all, welcome to Melbourne. Lovely to have you here. I guess with the post-decisional ERPs, that was interesting to see that pattern of the error positivity thing going, being quite sensitive to these change of mind measures for maybe objective errors, I'm assuming, compared to correct responses. And I think we also saw this in sort of post-decisional confidence ratings, and as it doesn't quite gel completely with some of these accounts, Kobe-December's general model. I was wondering what your interpretation of that was. Is there anything special about, error detection or something like that, or is it related maybe to these sort of ranges of confidence rating or change of mind ratings you might get sort of in those conditions?
Yeah. Just a short side remark, you're completely correct to kind of sort of conceptually confuse confidence and change of mind ratings, because essentially we believe it's the same process. The methodological thing that we introduced to kind of highlight changes of mind was that changes of mind were behaviorally relevant, so people could earn points if they changed their mind. So, which I didn't highlight in the presentation. Now, in terms of this potential kind of post-decisional, say, error-related positivity, it behaves a lot like CPP, so it seems to kind of reflect some sort of process of evidence accumulation, I believe. But what we are showing here is that, well, broadly speaking, we believe that that process that happens after people have pressed the initial response button actually reflects a different type of evidence accumulation, which would then be some sort of evidence accumulation in gauging whether you were correct or wrong, which is not necessarily the same evidence accumulation as your initial kind of accumulation, whether something is left or right. These are two conceptually different decision kind of processes. Having said all of that, if these are two completely different decision processes, then these discrepancies between pre-decisional and post-decisional kind of correlates or with confidence or metacognitive judgments potentially have some sort of explanation in there being two different cognitive processes. So that's my current kind of take.
Thank you.
Yeah.
I've got a question for Kelsey.
Yeah.
I didn't get around to asking this in the first session. I'm curious to thinking about your conceptualization of the self within a predictive processing model. I like the sort of the schematic you showed of sort of the feedback loop. What I'm wondering about is what your thoughts are on how you might integrate within that model kind of people's preferences over their own sort of self-attributes. So, you know, let's say I would prefer to be a kind person. I would prefer not to be a cruel person. I would prefer to think that I'm good at gardening, to use your example. I would prefer not to think that I'm bad at gardening. Is there a natural way to incorporate those preferences into a sort of error correction process?
Yeah, and I think that that also helps drive how you solve the errors that you do experience. So if you have a strong preference to be a good gardener, it's very socially valuable. There are lots of other contextual pressures on the choice about whether to abandon that as a self conception or whether to change your behavior. There's also the question of whether we have insight into all of the errors that we're receiving and at what level of the hierarchy we're receiving the errors, and some of the things might be more susceptible to change and easier to change, and some things are based on priors that are longer into our history and more embedded and harder to change, and, you know, and therapy is good for those sorts of things. But like, yeah, I think that that comes into you have options for dealing with the error, and you might be biased towards particular options because of all sorts of contextual factors, including social and personal values and what you want to be true of yourself.
A question for Robin. Can you tell us a bit, in as simply as you can, why your method seems to be much more precise for finding the target region compared to previous method? And what's the kind of approach?
Yeah, sure. I mean, the answer is around the computational approach. Yeah, it's, I mean, one of the previous methods, basically they looked at brain connectivity between the subgenual cingulate region and the dorsolateral prefrontal cortex, aiming to determine an optimal target within the DLPFC. But they basically just looked at which voxel in the DLPFC was most anti-correlated with the subgenual cingulate cortex. The problem is there's like a lot of noise in fMRI. So looking at a single voxel is not likely to give you a very reproducible result. So our approach was basically one aspect of it is just generating clusters. So seeking identifying clusters in the DLPFC that have like a certain brain connectivity with the subgenual cingulate cortex, and then just targeting the center of gravity of that cluster. And that is like really very reproducible. So that's part of it. And then the other aspect is, it's technical, but like the subgenual cingulate cortex, it's only around like 20 voxels in the brain. And it's also in a region of that has signal dropout. So basically, like the signal is very weak from that brain region, or it can be at least. And so one of the approaches is to generate it's called a seed map. It's just like a brain connectivity network based on the subgenual cingulate cortex. You do that in like 1,000 people, get the average, and then you use that as a proxy of this signal from the subgenual cingulate cortex. It's a little bit, I mean, it's a little bit, it's not complicated, but it's just like, it's probably all you'd want to know. Yeah.
Hello.
I have a question to Dragan. So about the sensitivity to the uncertainty.
Yeah.
So there, like you measure neural response and behavioral response.
Yes.
So which one has stronger impact, impact like uncertainty have on neural responses or behavioral responses? So I want to compare between, effect of uncertainty on neural responses and behavioral responses.
Presumably, the first impact should be observed in the neural responses because they precede the actual responses, behavioral responses. So in that sense, my reasoning is that the certainty or uncertainty of the decision should be observable in the neural responses. I'm not quite sure what do you mean by behavioral responses, because in these particular studies, we had two behavioral responses. So there's the initial response, and then there's the kind of change of mind response. So which of those two do you have in mind?
Change of mind.
Change of mind responses. Okay, and well, what we were showing was the relationship between neural responses or how neural responses predict change of mind responses, and whether that's modulated by our parameters that presumably should index or modulate decision difficulty, and by implication decision uncertainty. I'm not sure if that addresses your question. I'm feeling I'm potentially missing something here.
I was just want to know whether you compare and how they're is the sensitivity is driver for both of them, or is
we actually have done similar analysis, if I understand you correctly. So there's a whole different way you can analyze neural responses in characterizing how strongly the brain is representing those motion signals. You can build a kind of you can extract a way to represent neural signals that are very similar to those probabilistic distributions that I was talking before. And we do a lot of those analyses. We call them feature-specific analysis, or forward encoding models, or you name it. There's all sorts of flavors of that type of the analysis. So we originally actually were quite interested to see if we can extract that neural signature, whether that will then modulate concurrently both those decision-related signals that I was showing today and behavior. Bad news are that due to paradigm constraints or whatever, we were not really able to extract very strong feature-specific responses. So I cannot really tell you how that behaves in this particular paradigm. I can comment on some other findings, but potentially that's beyond this scope.
Okay, thanks.
Thank you.
Okay, I think we'll take one last question.
For Natalie. So the correct signal to correct your priors would have to be, you'd have to boost the signal of the absence of pain, which is what is happening in healthy individuals. But the problem is the absence of pain is by definition no signal. So is there a way to do that, get a signal in that's analgesic, that would fix your priors basically?
So I'm not exactly sure about the specific question, but I'll attempt a response. Basically, what you're saying is that there is no signal if there is no, for example, drug. Is this in relation to the first?
Interference, you said. So you said people with persistent pain or chronic pain still get pain despite not having a sensory input.
That's right. So basically the idea there is that potentially people with chronic pain are relying more on priors and less on the sensory information. One way of approaching this would be to try and change their biases and affect them with respect to their priors. But one other potential avenue is to look at whether their sensory information is somehow not attended enough, and therefore come up with some sort of interventions that would target that, make people more focused on their senses, see whether they can shift the processing of the overall percept towards the senses that they're experiencing at the moment. Presumably, if they no longer have the chronic pain generator that originally started this journey, attention to how much better they feel now compared to then would be a way to make them feel better. So basically, showing the distinction between the two is probably an important first step to see whether we can come up with new interventions that could help potentially chronic pain patients. I hope that answers.
Please stay for a moment, and please join me in thanking all of our speakers from this morning. And now I'm going to hand it over to our director, Marta.
Okay, you stay there. So I'm so pleased to see such a full house, and I really hope you all come back after lunch. Lunch will be served downstairs, again in the same spot, I believe. Yes, Sarah says yes. So do come back. I want to thank the chairs, Iroise and Sarah, for gracefully and assertively keeping us all on time. So they've done a wonderful job. And of course, to our speakers, they've done such an amazing job. All the talks were so engaging and inspiring, and you've all been so engaged. And I just hope that the same happens in the afternoon. I'm sure it will. But before you all go away, I want to give some little recuerdos. Thanking you for being so inspiring and generous with your time. A little bit of our culture.
Good. Thank you so much.
Yes. So thank you. Right. See you all soon, and we'll be back at 2 o'clock. So see you all back at 2.
Start the afternoon session, and welcome back to the Cognitive Neuroscience Annual Symposium. And hello everybody, welcome back to this afternoon session. I'm very happy to invite the panel of 3 speakers this afternoon. We've got Associate Professor Kate Hayward, Associate Professor James Coxon, and Dr. Daniel Bennett. And then we'll have a panel discussion. So first up, I'd like to invite Kate to come up. So she's an associate professor in the Department of Physiotherapy. And she also works at the Royal Melbourne Hospital. And she works on stroke recovery and rehabilitation. And she holds significant fellowships, including the NHMRC Emerging Leadership Fellowship, the Heart Foundation Future Leader, and a Dame Kate Campbell Fellowship as well. And she serves on international stroke recovery guideline initiatives. So it's my great pleasure to welcome Kate to this talk.
Thank you. Hello all. You have had beautiful sunny Melbourne weather come on for you. Feels like first summer day for all of us. So I am oh, that does work well there. I think that's nice and loud. I have to begin by fully acknowledging that this talk that I'm providing on the Parsi Zodiac trial is on behalf of Professor Amy Broadman. Amy was the principal investigator on this trial and is based at Monash University, and it would not at all have been possible without Amy and all of her efforts. Funding sources are listed there. But like all clinical trials, some of you might know Jeff Donnan. He calls them a contact sport. There are a lot of people involved in clinical trials, so many acknowledgements there up front. This way? No? Amy is front and centre there if you don't know Amy, and then all of the amazing people that made this happen. So many thanks and shout out to all of those people. So I would also like to acknowledge that all the work done in this trial was on the lands of the Wurundjeri Woiwurrung people of the Kulin Nation, and I acknowledge their elders past, present and emerging, and any First Nations elders in the room viewing this presentation. So just a snapshot to sort of set the scene before I dive in, the importance and the rationale for why we did this work. So stroke does increase the risk of cognitive impairment, and dementia is also without proven prevention strategies. But we do know that doing cardiorespiratory exercise can preserve brain health generally in our population, with limited evidence though in the context of stroke and dementia at this stage. And then cardiorespiratory exercise is recommended for people after a major cardiac event, but we don't have a lot of clear prescription guidelines in the context of stroke. So Amy led a study called Canvas, which was an observational study that got people very early after stroke on the acute stroke unit, as you can see by the first time points there on that graph, and then serially imaged them out to a year post-stroke, and was really interested in measures of cognition, that's Amy's key area, as well as the brain changes that occur in people after stroke. And these were all people with an ischemic stroke. And so on the y-axis, you've got your hippocampal volume, and as I said, time on the bottom. In the top line is your healthy aging people, and then we have our stroke populations underneath. And you can see that there's this decline after stroke that happens really in that first couple of months after someone's had a stroke, where they have quite a marked decline in their hippocampal volume. Other work that came out of Canvas was looking at physical activity after stroke. So they had people report how much physical activity they did, and they did see changes in brain networks associated with that. And that's the study on your right-hand side. No, I'm going to get my lefts and rights wrong. I'm not very good at that at the best of times. And then on the other side, they did some work as well at looking at physical activity and the development of white matter hyperintensities, such that people who did more physical activity that adhered with the guidelines actually had less white matter hyperintensities. So all of that really comes together and provides some of the rationale that feeds into this here about what the benefits of physical activity and exercise are for brain health after stroke. So we know that they're increased BDNF, we have increased VEGF, increased hippocampal volume, and you have these cerebral blood flow changes as well. And can that result in improved cognitive function and reduced dementia risk for people after stroke? We didn't know before doing this study, and that was the question that we were asking here. So we sought to determine whether cardiorespiratory exercise intervention preserved hippocampal volume and cognition in ischemic stroke survivors. And we hypothesised that cardiorespiratory exercise intervention participants would have less hippocampal atrophy at 4 months, so that was post-intervention, than a balance and stretching control intervention, so it was an active control. So the study design was a phase 2B, randomised open label assessor blinded controlled trial. And they were randomised to 8 weeks of balance and stretching. And that was our control intervention. They wanted an active control because it was perceived that if people felt that they weren't getting anything, recruitment could be quite hard. Or the cardiorespiratory exercise intervention. Now, we identified people very early post-stroke in partnership with many of the stroke units here in Melbourne, and identified people who might be eligible, and then would contact them to start at that time point of 2 months, reflecting that change that was observed in hippocampal volume early. And then there was 3 study visits, so that baseline at 2 months, post at 4, and then 12 months as a follow-up. Now, Pisces had the great joy, we can say now, of operating within the pandemic period. So initially it was an in-person intervention. They were coming into the outpatient facilities at the local hospitals and doing the therapy there. Obviously, when COVID hit and Melbourne particularly experienced many, many lockdowns, we had to shift, and it became a Zoom-delivered intervention that people actually did from their home. I think this may be the only study ever in my career where we had a storage locker so that we could store the bikes there, so that we didn't have any cross-contamination with blinded assessors, and they got delivered to people's homes and all of that. So a big shout out to the entire team there who made that possible. Who did we include in this study? So as I said, confirmed ischemic stroke, adult stroke survivors. They had a pre-morbid modified Rankin of less than 3. And what that means is they were relatively independent out in the community, and they were able to do things like their finances themselves, get about, manage all of their activities of daily living. They had to have good English, and that falls in with lots of the cognitive measures, and then able to nominate a support person, which was an addition when we flipped to that Zoom delivered at home. And then the exclusions, there was several around the MRIs that we wouldn't be able to take some people because of that there. So this is an overview of what was collected. As I said, identified people very early post-stroke. That 2 to 4 months was when the exercise intervention occurred. They did have check-ins at 6, 8 and 10 months post-stroke. And that image at the top shows that beyond the clinical measures, there was quite a lot of other measures collected. Now, the analysis for this study only finished early this year, so all of those additional measures are still being analysed. You can see that there's stool samples there, blood. There was a lot of different measures that were collected with activity monitors, and then the imaging scans can be used for many other purposes as well. So a lot of that work is ongoing. This is an overview of the intervention. So it depended on what group the person was in. So you can see the green on the bottom is the control, and they did their balance and stretching. So they're still getting up and doing things, but they're not really getting their heart rate up. They're not really having that exertional piece there. The blue on the top half is the intervention component of the cardiorespiratory exercise. This had a progressive element to it so that you could get people going in that preparation phase, because some people may not have been very big exercises beforehand, and there can also be quite a bit of fear after you've had a stroke about getting exercising, getting your heart rate up and those sorts of things, and then progressing the intensity as they move through it. Now, everything was always observed by a trained exercise professional. Even when it was Zoom, there was a trained exercise professional for all of the intervention sessions that were provided 3 times a week, 60 minutes in duration, and there was always a warm-up and a cool-down. So the primary outcome measure that we were interested in this trial, and this is what we were powered to look at the effect of, was relative hippocampal volume change from that 2 to 4 month period post-stroke, so the intervention period. The secondary outcomes of interest, we're looking at that longer term time point. So the 12-month executive function using the trail-making B test, looking at that in time for the seconds. And this was adjusted for time at baseline and their modified Rankin, so that measure of general ability after stroke. We also looked at TBV, so total brain volume changes over the time points as well. We had safety outcomes, so we always have those within our clinical trials, and we had key ones of death and then other serious adverse events related to the intervention. But we had some special adverse events of interest as well. So we looked for stroke and TIA that might have occurred during it as well. And then a lot of tertiary exploratory, which we are not powered on any of those besides the primary. The statistical approach, so we had 90 participants per group. When we did make the switch to going to a Zoom delivered, we increased the sample size then as well, and our blinded statistician did all of that. So we did also increase the possibility that we would lose participants. Obviously with COVID, people mightn't be able to do the full 8 weeks, and other things come into it that might have impacted. The randomisation schedule was stratified based on total brain volume and that modified Rankin, 0 to 1, very, very mild, and then the 2 to 3 based on their pre-morbid level. And then we did a modified intention to treat approach them. And that's because of the way that the trial was run, is that you had to consent people, then do all of the imaging aspects to make sure that they could go in the scanner, and then you might find that they're not. So they actually never had any intervention. So that's where our modified intention to treat comes in. There is some subgroup analyses that I'll show as well. So this is the flow chart of participants into the trial from a very big number screened. And if you haven't done trials on clinical, it always looks like this. That's because they're looking at every single person that comes through a stroke unit. Somewhere like Austin has around 450 to 500 stroke patients come through each year versus a Royal Melbourne's about 1,100 stroke patients. So you can see if you're looking at every one of those people, your number's going to go pretty high. Once we flow everyone through, it came down to 49 people in the intervention arm and 55 in the control arm that were treated. And there was no one that died in it, thankfully, but we did have a couple of people who withdrew before treatment or had to discontinue for various reasons. Here's the demographic, so relatively young population for a stroke trial. You can see that we had a lot more males in both groups, and this is very common in stroke trials and especially rehabilitation and recovery trials. there's many people trying to look at how we can do that a little bit better. You can see there modified Rankin, so pre-stroke was 1, so very, very mild. And then the NIS is also highlighted there. So that's the National Institutes for Health Stroke Severity Scale. It goes up to 42, so a very, very mild cohort of people that were enrolled in this trial. And most people, it was their first clinical stroke as well. This is some of the baseline measures around their brain volume and their total stroke lesion volume there. So they're very, very small stroke lesions, and this is the image here of what the lesions look like for the cardiorespiratory exercise on the top and the control intervention on the bottom. So this is condensing everyone all together, obviously, so they look a little bit bigger, but generally they were very, very small lesions. The groups did have some difference in their baseline vascular risk factors, particularly atrial fibrillation. So the control group had a lot more people with atrial fibrillation, but then the exercise group had a lot more people who had type 2 diabetes. Baseline cognitive status. There's a few different measures that we used within this. Our main one of interest was obviously the trail making test, and we had 100% of people able to do that. And the time to complete, the control arm were a little bit faster at baseline than the intervention arm. Just to cover off the intervention that was provided before we get to the primary results, is you can see that people did achieve, so it was 8 weeks by 3 sessions. So they're getting up there. So the median number for both groups was around 20. So they've missed a couple, not uncommon. People have appointments and things. It can always be hard to keep everything going. But the number of minutes with the trained exercise professionals was comparable between the groups, and the time spent in that cardiorespiratory effort phase is reported that only for the cardiorespiratory exercise group, because obviously balance and stretching, that we weren't monitoring that from that perspective. When we look at the primary outcome here, so this is hippocampal volume, and we're looking at the change. We can see that there is no difference between the groups at that across the pre to post intervention period. When we look at the secondary outcome, so we look at the 12-month trails making B, we do actually see a significant improvement in trails B when it's adjusted for baseline modified Rankin. So when you look at the raw, you go, the control is actually faster, but when you adjust, it's the intervention arm that is almost 4 seconds faster. We're not powered for that at all, though. So that's what you have to remember when you look at that. But it appears like people were a little bit faster there. From a safety perspective, as I said, no deaths. Serious adverse events were comparable between the groups, and recurrent stroke, there was 1 per group. So that is a really, really encouraging outcome as well, that people are able to be very, very physically active. Oops, gone back. That was the 5-minute one, wasn't it? Yeah, perfect. And then the other encouraging part from the cognitive side of things was that the ADAS COG score also went in the same direction when adjusted for that baseline modified Rankin, in that it was in favour of the cardiorespiratory exercise intervention over the control as well. Again, not powered for this, so we do have to be mindful of how we interpret that, but it was interesting to see both of those effects on cognition. There was no real differences when we looked at anxiety, depression, quality of life, and fatigue. Yes, that looks better on yours than the one there for me. So from a sub-analysis, there wasn't really anything of interest to come out there. There wasn't anything different between subgroups of interest to give us any other indication around the findings, seeing that they all cross that midline on the forest plot. So what this tells us is cardiorespiratory exercise is safe when delivered at 2 months post-stroke. You might think that sounds really simple, but it actually isn't very commonly known. And as I said, people do have a lot of nervousness about getting back to doing it. It did not preserve hippocampal volume more than a balance and stretching control, and it may benefit cognitive preservation up to 12 months following ischemic stroke. So here, if we look at the findings and in light of those Canvas findings that I showed you earlier, you can see that the people enrolled in this trial were different at baseline from the Canvas group. So they had a different hippocampal volume at baseline, but we don't see that decline occurring in the group. So there may be something here, but it does need to be looked at in much more detail in follow-up trials where that was the primary outcome of the study, but encouraging findings. So as I said, safety, really important that we know that people can do this and it can be delivered remotely. Does maybe say that whatever you get out there and doing could have a benefit on you for your brain as well as your cognitive function after stroke, which could be really encouraging for people who don't like doing really high intensity exercise, that it's about getting out there and doing something, because a lot of these people weren't very physically active beforehand. So it's a very big behaviour change to get them to think about that. And then from a brain health, we need to think about how do we integrate this in for prevention and successful ageing. And it does indicate that likely we need more trials that are powered on the cognition aspect as opposed to the brain changes. So this was published in JEMMA Network Open in late August. And there was a very nice editorial written as well by people in the US about it, just highlighting the novelty of the findings for the field that hopefully will spark a lot more studies to understand cognition and brain health in stroke. Thank you.
Thank you very much, Kate. That was a beautiful talk. Thank you. I'll get you to take off the mic and, oh yeah, I'll give it to James. So, thank you. Okay. So here comes James. So James is a professor at Monash University, associate professor in the Movement and Exercise Neuroscience Lab in the psychology department. And his lab works at the interface of cognitive neuroscience, neurophysiology and exercise science. And his research explores how cardiorespiratory exercise, it's a theme today for this afternoon appears, influences neuroplasticity, learning and cognition. So welcome, James, and got that going.
So, okay. How about now? Yeah, good. Thing is, I'm not going to talk about exercise today. We just got it. We know it's good for us. But yes, the lab has been doing a lot of work on high-intensity interval exercise, including in older adults over the last few years. So be sure to have a look at the recent outputs from the group in that regard. But that was a great talk. I really enjoyed it. But today I'm going to talk about another line of research we've been doing, which relates to the offline brain. So when we, in the initial stages of learning a motor skill. So we're really interested in how do we go from this to this? And that's an experience pretty much everyone here should have some experience with when they wrote their PhDs or any other kind of thesis. That, at least for me, I started off rubbish. By the end of my PhD, I was good. So with practice and time, I was able to acquire skill. We know that a lot of the brain is into learning new things, learning new skills, right? So motor skill learning is not simply something that relates to your primary motor cortex, but you've got a lot of the brain getting involved in different aspects of skill learning. And also there's data and theory out there that implicates different brain regions being differentially involved over the time course of learning. So areas like the prefrontal cortex, associative territory of the striatum, and the hippocampus are thought to be involved quite early on. And I'm going to come back to the hippocampus later in this talk. Okay, so this is a typical looking learning curve, skill on the y-axis with time, whether it's quite a simple skill like learning a task in the lab, like a little tapping task, you might acquire a reasonable level of proficiency in a matter of minutes, or other types of much more complicated skills that could take months or more to learn. In the lab, it's tractable to do tasks where the learning curve is pretty quick, but there's some other advantages to that, as we'll get into. And so we learn, and the traditional model is that we think we learn a lot while we actually do practice-dependent learning. And then, so we can have these gains as we practice, and then there's the time between practice when we're not, so offline learning. And during that time, you might have either time-dependent processes that take, say, minimum 4 hours for various types of consolidation to occur, gene expression, bedding in of new ion channels, all that sort of stuff. It takes time, and so that could help stabilize a newly learnt memory, make it more resistant to interference so you don't go backwards, so you're not starting from scratch all over again. Or you might even get some sort of little offline improvement, and sometimes, depending on what it is you're learning, some of that might be sleep dependent. So consolidation has traditionally been thought of over this much longer time frame of hours to days. But we're going to focus on this really early initial phase of learning, the fast learning phase. And this study was an important one for what I'm talking about, where they deliberately made the task involve alternating epochs of 10 seconds of task, 10 seconds of rest. It's a pretty simple task, 4, 1, 3, 2, 4, tap it out as quickly and accurately as possible, as many times as you can, then take a break. And so that's what Bonstrup and colleagues had participants do, and they showed the typical learning curve, and that pretty quickly you get almost as good as you're going to get within that session. All right, so after even just 3 or 4 minutes of training, they're at about 95% improvement for the session. So that's that vertical black line. And what they did that was conceptually interesting and shed some new light on what the brain might be doing very early in learning was that they had this more granular partitioning of the skill improvements through those initial trials. So you can see these changes from the beginning to end of the short block versus the changes that occur from the end of one block to the beginning of the next block. And if you sum up those changes over all of the blocks, you get these micro online effects, micro offline effects. And when they focused on that and looked at these summary measures, the outcome was that the majority of the very early learning came from the micro offline gains, while the participants were not actively practicing the task. And they showed that in the lab, and then they showed it with a much larger crowdsourced online sample as well. Of course, when we learn motor skills, we can use our declarative knowledge. We can maybe have the sequence explicitly given to us. We know what we need to do, how to do it, and we focus on that. We can also have tasks and paradigms in the lab where we give much less information about what needs to be learned in order to try to tap into some of these more implicit aspects of how we learn skills. So on the example for implicit, it might just be that you see these visual cues and your task is just simply to respond as quickly and accurately as possible. Participant is never told of the presence of a sequence, so they don't even know it's there. And then you can probe them at the end of the experiment to make sure that they had very little awareness, which is how we run these tasks. And that's what we did in the first study from the lab that looked at this effect. So this was PhD work of Emily Brooks. Emily had a task with identical to the Bonstrup, 10 seconds task, 10 seconds rest. We never told the participants of the presence of a sequence, but there was a 12-item repeating sequence there, and through practice, people get better. So they're just told to be as quick and accurate as possible, responding to the cues. And then we probed them at the very end of the experiment and were able to quantify that they really didn't know very much about the nature of the sequence or that it was there. We saw the same sort of typical learning curve. With practice, people get better. And again, that the majority of those very early learning gains related to the micro offline periods. then did this again. This is the work of honours student Namin Al-Jahani, where in a larger sample, again, it was the micro-offline contribution that was doing more of the early learning effect. And in this study, we also collected EEG. So same task, just with EEG. And then with that data, that one hasn't transferred from from Windows to Mac to Windows. Task and rest. Okay, so we've got the 10-second alternating epochs, and we went from time domain EEG data into frequency domain by applying a Morley wavelet transform, and got the power modulations in the alpha, beta, gamma range. On the color coding there is the percent change in power relative to a resting state EEG scan that we acquired before doing the task at all. So just 2 minutes of complete rest. And so anywhere where you see blue, you have a reduction in beta power. There's a well-known event-related desynchronization in beta when you engage in movement. And then when you stop moving, beta resumes, it returns, and you get an increase in power, event-related synchronization. So we were particularly interested in looking at the beta modulations during this task. And what you can see is in the beta band, across most of our electrodes, we are seeing a significant modulation there. So reduced beta during task epochs that comes back during the rest epochs, a reinstatement of beta power. But the question that was interesting there is, to what extent does beta power come back from one individual to the next? And does that have any relationship to how well you experience these offline gains during the rest periods? And we're able to show that in a subset of these electrodes that when we looked at beta from those resting epochs, the lower that resumption, the weaker the resumption of beta, the higher the offline learning was. And what was nice about that is that it aligned very well with what others have shown with MEG, with the explicit version of the sequence task. And so again, lower beta power during those resting epochs, greater micro-offline gains. And beta can potentially relate to a few different things, but one of the things it might be indexing is aspects of inhibitory tone. For example, when we move, we have to take away all the inhibition for the movement to happen, and that's corresponding to when beta is disappearing or dropping markedly. So in another study, this one's recently published by postdoc Dr. Josh Hendricks, we looked at GABA with measures of from magnetic resonance spectroscopy, so looking at GABA concentrations in the motor cortex, and we did resting state fMRI. So same task, and the key outcome from this study was that a regression model that combined the motor cortex GABA concentration and the default network connectivity predicted or was associated with the magnitude of the micro-offline effect. So lower motor cortex GABA and stronger connectivity in the default mode network. And the default mode network includes the hippocampus, which gets me to some of the other work that's been done, some really exciting. This one's quite complicated. I couldn't necessarily tell you all about the methodology of how we went about this, but this is Ethan Book, again in Professor Leonardo Cohen's lab, using MEG and some parcellation and decoding. And what he found was that there was evidence in the resting epochs of a replay of the sequence from the task epochs. It was temporally compressed about 20-fold compared to actually doing the task. And then again, and that was for a principal component network that included the hippocampus, and that the rate of those replays predicted or associated with the magnitude of the micro-offline gains. Going further, this is a study from this year where making use of patients who are about to undergo surgery for epilepsy. Shurgard and colleagues had some EEG electrodes where they could implanted intracranial, where they could get signals related to the hippocampus, and they looked at ripple rates, with ripple rates being one of these electrophysiological signatures of replay. And they show, again, this association between greater ripple rates and a higher micro offline effect. So reasonable amount of evidence across these studies that on the one hand, there's something going on with beta modulations up in the cortex that could relate to micro offline gains, and down in the hippocampus, replay ripples as being potentially implicated. But a lot of this is this correlative evidence. And what we really need is some studies that try to look for causality. Can we show that the hippocampus really is a critical area for these very fast early learning effects when we learn a novel motor sequence? So that's something we've been doing this year. It's very preliminary data. It's actually an ongoing study, so I won't try to make too many strong claims about this. This is the honours thesis of Alice Safri. And in this task, in this particular study, we were using a new non-invasive brain stimulation technique called transcranial ultrasound stimulation. So we're sending ultrasound waves using a transducer that has multiple elements. It gets attenuated as it goes through the skull. We have to model all of that, do a lot of simulations. But then when the phasing of the sound waves comes into alignment, you get this amplification effect. Think about waves at the beach and when they come together and double up. Same sort of idea, or if you know about spatial audio and you've gone into a hi-fi shop and you've stood in exactly the right spot and all of a sudden you can hear the sound where you couldn't hear it anywhere else. It's that idea. But we're using it now to try and deliver sound energy, a mechanical pressure effect, deep inside the human brain. So quite low intensity. We do a lot of simulations where we model it to make sure that there's very limited thermal effects. With the simulations would suggest about half a degree of thermal rise at the level of the skull, but then way, way less inside the brain. Right, so this is a very different way of using ultrasound to how some others are doing it in the hospital environment, doing non-invasive neurosurgeries for thermoablations. We're orders of magnitude lower in intensity, trying to use it for neuromodulation. And the logic is that we have these mechanosensitive ion channels in cell membranes, and that by inducing the sound pressure, we can then modulate the state of those cells. And depending on how we pulse the stimulation, we can induce effects that might be excitatory or inhibitory. So we are trying to use a more suppressive type of ultrasound in this particular task, and we delivered it during those offline periods. So during the initial blocks of the task, in the offline epochs, we were delivering the ultrasound, and we either delivered sham, so just turn the machine down as low as it can go so that really no energy is getting inside the brain, but everything else is the same. There's still lots of squishy ultrasound gel everywhere. Or we delivered the real stimulation, and we did that in a double-blinded way for the data you see here. And then when we focus in on just the very early blocks, the first 8 when we delivered the ultrasound, if you look really carefully at the blue lines, you can see that where you end up compared to where you begin, the next block is going up. So it's this positive micro-offline effect in the sham condition, as we would expect from all of the studies I've shown up to this point. In the TUS condition, it's much less evident that that's what's going on. And so this micro-offline effect appears to be somewhat reduced. Now, like I said, this is an ongoing study. It's early days. We're probably going to have to keep going and look at more conditions and participants and further refine some aspects of our methodology, but I think that this is an encouraging start. So to sum up, hopefully through this talk, I've convinced you that when you first learn a new motor skill, it's not all about when you're actually trying, but taking little breaks in between attempts can be really important. and that the micro-offline gains account for quite a significant proportion of that very early initial learning. It doesn't really seem to matter if it's a task that weights heavily on the more explicit side of how you learn skills versus the more implicit learning. In both instances, the hippocampus may well be playing a role in aspects of bedding down those sequences. And so we've got a picture emerging of neural correlates that involve beta oscillations on the one hand, and perhaps reduced inhibition up in the cortex, and replay down in the hippocampus, and that when we target the hippocampus with ultrasound in an attempt to suppress what's going on during those offline periods, we've got some initial trending indication that it can reduce the magnitude of the micro-offline effect. So thanks for your attention. I'm at Monash. I'm also delivering a workshop the day before ACNS. So if you're attending ACNS and are at all interested in the technique, we're going to dedicate a whole day to what it involves. There's a lot of simulation of the pressure fields. There's neuronavigation in order to be able to track the right vector to get to the right spot. And so forth. And so come on out to Monash for the day and learn more about it if you're curious. So thanks to the sponsors and for your listening.
Thank you, James, very much. That was a beautiful talk, very clear. Thank you so much. Now I would like to welcome Dr. Daniel Bennett up to give his talk. So Daniel is a senior lecturer here in the School of Psychological Sciences, and his research group studies human reinforcement learning and decision-making with a focus on understanding decision-making in contemporary gambling products. And Daniel completed his PhD here in 2017 and then went away and did some fantastic work at Princeton and Monash, and now he's back again. So let's welcome Daniel here today.
All right, thank you for the introduction. So what I'm going to be talking about today is some kind of a bit of a summary overview of some work that I've been doing for the last kind of 3 or 4 years in collaboration with a whole bunch of different people who I will point out at the end of this talk. But basically the context for this talk is that I did my sort of research training in cognitive psychology looking at decision-making under uncertainty. And what I have been trying to do in the last 3 or 4 years is to sort of take some of those skills of basic experimental psychology and apply them to a domain that I think they're very suited to, which is contemporary real-world gambling products, and trying to understand why people gamble and why they make the specific kinds of gambling decisions that they do within the interfaces that are provided to them in these contemporary products. So in case I need to convince you that it's important to study gambling, here's one summary plot. So what I'm showing here is the monetary loss per adult per year in Australian dollars. So Australians in 2022 lost more than $1,500 per person. That's not everyone in this room losing $1,500. That's particular people experiencing very high levels of harm and then lots of people experiencing no harm. And what you can see as well is that Australia is doing this at far higher rates than elsewhere in the world. This isn't purely a cultural thing, even if we look at putatively culturally similar places like New Zealand, although James and some of my New Zealand family might disagree. We can see that it's not simply the case that something about being in this corner of Oceania is incurring this much harm. It's something specific about what's going on in Australia. So this is part of the motivation for doing this line of research, is saying something is going on in Australia. There's something about the ways that Australians are making gambling decisions that are leading to high levels of harm, and let's try and understand what that might be. Now, there's lots of different places you can take that line of research, and we're interested in pursuing many of those avenues. The specific feature that I'm going to be talking about today is a feature of contemporary online sports betting that has kind of come to prominence in the last 10, 15 years or so. You'd be very familiar with it if you placed a bet within a sports betting app. It's called instant cash out. Now, the feature is probably easiest explained through some of the marketing that gambling companies use. So what I'm going to show you here is an example of an advertisement for instant cash out that relates to this English Premier League game on the left over here. So this is one where, I don't know if you can see, Manchester United is playing Everton in 2012. Manchester United goes up 3-1, and then Everton comes back to draw the game. And this was very consequential. I think Manchester United didn't win the Premier League this year as a result. And the bookmakers then made an advertisement referencing this game, which was sort of prominent in popular culture at the time.
Looking a little vulnerable. You've backed United to win. Do you cash out or don't you? Do you? Don't you? Do you? Cash out from Betfair lets you change your mind during the match. Go to betfair.com/cashout. Don't settle for less.
So what's being advertised here is the ability to change your bet after you've already placed it, in this case, so that you're the only clever one in the pub who's made some money back and everyone else has lost money. All right. So what's basically going on here is that instant cash out is a feature which allows people who have placed a bet on a particular event to cancel that bet at any time during the event in exchange for an instant payout. Now, the way this technically works is that the amount you're offered to cash out sort of increases when your bet is more likely to pay out and decreases when your bet is less likely to pay out. More formally, we might say that the cash out offer is proportional to the expected value of the bet. Now, why might bookmakers offer this feature? It's not out of the goodness of their own hearts, it's because they make money out of it. So cash out is typically offered to consumers at disadvantageous prices. So what that means is, if you look at the example up at the top right here, our bettor is being offered £27.18 to cash out. What we know from the way that these offers work is that that probably means that the bookmaker estimates that the value of that bet at that point in time is £30. And so what that means is that if somebody chooses to cash out, yes, they get £27, but the bookmaker has sort of made £3 in expectation. Even though this is the case, and it's sort of fairly widely known that this is the case, cashing out is a fairly popular feature of sports betting apps among people who place bets using these apps. And it's kind of not hard to intuit why this is. And if you were confused about this, you would just have to look at some of the other advertising that bookmakers offer. So here's a selection of advertisements of the cash out feature. So what you'll see if you look at this is there's sort of some fairly consistent themes coming through to do with taking control of bets and being in control of bets. Now, various industry bodies are at pains to say that they are not advertising cash out as a way of responsible gambling. They just happen to be advertising that you can be more in control if you happen to use this feature. Okay, so there's an applied rationale for studying this, which is what we know from a couple of different studies, including some that we've done ourselves, is that people who use the instant cash out feature more frequently tend to experience higher levels of gambling-related harm. So this is, formatting slightly bodged, but basically if we look at people who regularly bet who experience higher levels of harm, and then a demographically matched group of people who regularly bet who experience lower levels of harm, and we ask them how often do they cash out, what we see is that people who experience higher levels of gambling-related harm tend to report using cash out more frequently. So this is, in spite of the fact that it should allow people to exert more control over their betting, what we find when we actually look at the frequency of these behaviours is that it's associated with greater gambling-related harm. There is also a sort of a theoretical rationale for studying instant cash out, which I'm going to go over slightly more briefly. But in essence, I can sort of illustrate it to you by showing you a hypothetical gamble. So let's say I'm going to toss 2 coins, and I tell you that you're going to win $75 if both of those coins come up heads, and you're going to lose $25 otherwise. And I ask people whether they would like to accept that gamble. Now, that's a sort of a decision problem of the kind that is fairly well studied in cognitive psychology. And the way in the field that you would sort of appraise something like that is you would say, okay, if I accept the gamble, then there's some probability of winning. It's 0.25 in this case. There's some probability of losing. It's 0.75 in this case. And I know that the outcomes are associated with each of those, and I know their associated probabilities. And so what that means is I can sort of calculate the utility, the expected utility of that gamble. I can decide whether to accept it or not. The interesting thing about this from a theoretical perspective is that as soon as you add a cash out option there, where I toss one coin and then ask someone if they want to cash out or not, and then toss the other coin, it becomes a much more complicated gamble from a theoretical perspective. So, okay, if the first one comes up tails, you lose for sure. But if the first one comes up heads, I might then ask you if you want to cash out. You might decide with some probability to cash out for some cash out offer. You might decide not to cash out. I'm not going to walk through the tree here, but the main point I'm trying to make here is that adding a cash out offer into a gamble like this one transforms it from being a relatively well understood, well studied decision problem into being something that's much more complex and it's much harder for people to calculate the variance of, to do all the things that you might want to do to sort of work out if you want to accept the gamble or not. So in that spirit, what we're going to do here is we're going to talk about the various ways in which the availability of a cash out offer within a betting environment influences the kinds of bets that people make. So this is the specific decision-making task that we're going to use to kind of illustrate this. It's a little bit complicated, so I'll just walk through it in a bit of detail. So what happens here is in this task, as a participant, you're told that you are you're either predicting. Sorry. The number 8 on the left is an 8-second timer that will start as soon as you click on the timer. So it'll count down 8, 7, 6, 5, 4, 3, 2, 1. And then at the end of that 8-second period, one of these cards is going to be chosen at random from this set of 10 cards. So it'll either be a blue card that's chosen or it'll be a red card that's chosen. And then what we ask people is to predict, before they've seen this, what colour the card is that will be chosen at the end of the delay. So what we're trying to do here is to create a kind of relatively controlled environment that replicates some of the features of sports betting. So you have sort of a bet that people are placing, you have an unknown outcome, and then you have a delay prior to the outcome within which we might be able to offer people the chance to cash out. So what people do is they first of all predict whether either the red card or the blue card will be the last one remaining. They then crucially decide how much they want to bet on that prediction. So betting less here is incurring less risk. Betting more is incurring more risk. And then we, on some trials, we offer them the chance to cash out during that delay period. So here you might see that if you look up the top left, the person has predicted a red card and bet 50 points. The timer starts to count down. We offer them one chance to cash out. You probably can't quite see that here, but it's sort of saying you can cash out now for 70 points if you would like. If you accept the cash out, the cards disappear and you wait for the timer to run down. If you reject the cash out, what happens is that you you win or lose points depending on whether your original prediction was correct or not. And then, actually, I've got a little video of this here just to kind of illustrate what this means. So here, the person is going to predict a blue card. They're going to decide how many points they would like to bet on the card being the final card chosen being blue. And then they'll start the timer. It's 5 seconds here just to sort of speed through the example. They offer the chance to cash out. They decide not to cash out. And then they, in this case, win because their prediction was correct. On the next trial, they decide to bet 50 points. Here it looks like they've made a bad prediction because there's more red cards. And now when they are offered the chance to cash out, they choose to do so. Okay, so hopefully that illustrates the sort of the kinds of decisions that people are making. Our primary research question when we ran our first study using this task was just if we have some trials in which people know in advance that they will be able to cash out, and some trials in which people know in advance that they will not be able to cash out, how does their betting behaviour differ between those two things? And what this is asking is, how are people behaving differently when they know that cash out is an option that is available to them? And our dependent variable is just going to be the number of points that they bet on the outcome of the thing that they've chosen. And so what we see across a couple of different replications in different designs is that when people know that they will be able to cash out, they are placing substantially larger bets. So this is really interesting because what it says is that despite the fact that people perceive cash out as being a sort of a safer or more in control option, when they are actually offered bets in which they have a chance to cash out, they are incurring more risk. We can also ask how do different people differ in terms of how often they cash out of their bets? So this is another study that we ran using this task where here, rather than looking at how much they were betting on the outcome of that card, we just looked at how often they were cashing out. And we looked to see how this was associated first with how impulsive they self-reported being. I'm using a factor analysis of a battery of different impulsivity measures, and also the extent to which they experienced problem gambling, which is this problem gambling severity index, or PGSI scale. And so what we found, which is quite interesting, is that the people who are cashing out the most frequently within this task are the people who are less impulsive. So the likelihood of accepting a cash out offer is greater if you are someone who self-reports being less impulsive. Similarly, if you are someone who self-reports experiencing lower levels of gambling-related harm, it's likely that you are cashing out less often in this task. With the caveat that we didn't have huge numbers. This is just a community sample, and so we don't have huge numbers of people with really severe levels of problem gambling. There's a few different ways to think about this. But one thing that I think is quite striking is that what these figures suggest to us is that in terms of the sorts of people to whom, the sorts of consumers to whom cash out might be appealing, it's not likely to be the rusted on sports bettors who've been doing this for 10 years. It's likely to be the people who might not otherwise have been attracted to the idea of placing a bet. And so this suggests that there are two reasons why cash out might be seen as a particularly lucrative product for gambling companies. The first of which is it encourages people to place larger bets. And the second of which is that it may mean that gambling as a whole appeals to people who might not otherwise have been enticed to place bets. And then lastly, what I'm going to do is talk about one more study. It's a recent work in progress, and so there's more to be done with this particular study, but it's sort of trying to test that hypothesis that's suggested by the advertising, that people like to cash out because they like to be in control of their bets. So what we're going to do here, and this is work by Andrew Kamara, who is a PhD student both at Monash and at Warwick in the UK. We are going to do a variant of that first study that I talked about, where we kind of manipulate the availability of cash out. So we can have cash out being available sometimes and cash out being unavailable sometimes. And then what Andrew has done, which is very clever, is to add a third condition where cash out is sometimes automatic. So what this means is that sometimes you'll be shown something like this. So first of all, you place your bet and you're told that cash out will be automatic. What that means is that rather than having control over the cash out decision, you'll sometimes be shown a cash out offer like this one and be told, okay, it was automatically accepted by the computer on your behalf, or it was automatically rejected by the computer on your behalf. It was accepted about 40% of the time and rejected about 60% of the time. The reason for that is because that roughly matches the frequencies with which people cash out when they can sort of freely choose to do so within this task. And then, so the logic behind this is that if the rationale or the motivation underlying people's decisions to bet more when they can cash out relates to their ability to control what's going to happen, then taking away that control by making cash out automatic should abolish that effect. And that is indeed what we see. So this is formatted slightly differently, but the basic effect is the same. So on the left, we have how much people bet when they know cash out will be available. On the right, we have how much people bet when they know cash out will be unavailable. So we replicate the basic behavioural finding, which is that people bet more when they know they can cash out. But the interesting new development in this particular project is that when people know that cash out will be automatic, that is, it will be accepted automatically some portion of the time, the amount they bet looks much more as though cash out were unavailable. And so what this kind of tells us is that part of that motivation to bet more when cash out is available is not just because it reduces the variance of outcomes or makes things more predictable or something like that. It's that it allows you personally to exert control over what's happening. And so this suggests that perhaps those gambling companies are onto something when they market this using control, because when we take away control, we take away one of the effects of cash out on behaviour. Okay, so I'll really briefly summarise what I've talked to you about here. What I've told you is that in a controlled behavioural task that we have designed basically to replicate some of the features of sports betting while under a greater degree of experimental control, what we find is that when people know that they will be able to cash out of their bets, they tend to place larger bets. I didn't actually show you the data. I cut this one out, but I can assert to you that one reason why people might be doing this is because it might help them to avoid losses. If you have questions about that, please ask me. It seems to be that instant cash out is predominantly appealing to less impulsive users of these products. And this has led us to a hypothesis which we are kind of entertaining at the moment and which we are planning some future research to address using a longitudinal study, which is that it's possible that this might mean that something like instant cash out could be a kind of a gateway product that appeals primarily to people who haven't previously used gambling products, sort of gets them in, gets them comfortable with the platform, and then so may lead to kind of an escalation. Because that would sort of explain this interesting discrepancy that we're finding where in our community sample, the people who are cashing out the most tend to be the people who are experiencing less gambling harm. But then when we look at a sample of people who gamble regularly, the people who are cashing out the most are the people with high levels of gambling harm. So there's a few different ways that we might kind of resolve that apparent contradiction, some of which involve methodological criticisms of our tasks. So I'll acknowledge that those exist and sort of say we don't prefer them. But another sort of possible explanation is that when we sort of cross-sectionally get people at different points in time, depending on how often they gamble, we are seeing these different associations between how impulsive they are, how much gambling-related harm they've incurred, and how much they like using this cash-out feature. And then we have some interesting work in progress suggesting that the decision to cash out is perhaps at least partially explained by people's preferences for being in control of what's going to happen. Now, this is work that has been done with a bunch of different people, a bunch of different colleagues and students down at the bottom left, and then a few international and national collaborators. I want to thank them, thank those in the room who are working on this stuff, who have come along. Thank you, guys, and thank funding sources as well.
Thanks, Daniel, so much. It was a great talk. Well done. Now, I'd like to invite James and Kate back up as well, and for you to stay here too. And we'll. Oh, yes, you could sit in the chair if you'd like. That would be lovely. Yes. Yes. Would you be happy to sit in one of these chairs? Okay. Hop in. And I'll open the floor up for questions. Yeah, do you want to? We can move it around if you like. Yeah, do that. Oh, yeah. James is going to be standing.
Although that was fun to watch.
No, that's great. Okay, so let's open the floor up for questions.
Thanks. Amazing work. Question for Daniel. So it seems to me that you are essentially demonstrating that it's about locus of control, really, and sense of agency. Have you looked into other behaviors that are impacted by sense of agency and sort of trying to correlate those gambling harms? So I'm just trying to kind of clarify whether this generalizes beyond the gambling context and to which extent is whatever you decide characterizing here specific for the gambling. Obviously, the harm is the most intense, but yeah, there could be other things that are impacted as well.
Yeah, really, really interesting question. I wonder whether you have something specific in mind, and maybe we should talk about that later. Andrew, the PhD student, is obviously really interested in control. I'm actually supervising another PhD student who's interested in desire for control. And one thing that I've learned from working with her, that's Laura Forbes, is that this word control means a lot of different things to a lot of different people. So it can mean how much you want to, just be in control regardless. It can mean how much you want to use control to avoid negative outcomes. It can mean the belief that, so this is what you're referring to, I think, with illusion of control, that just things just go better when I'm in control of things. And so in response to your specific question, we haven't looked at how this correlates with other sorts of behaviours that are associated with the illusion of control. But we're sort of thinking, Laura has been developing a task that sort of tries to measure how much people want to be in control. And there's a planned project for next year, which will be sort of looking at whether people who want to be in control more show this effect more strongly. It'd be interesting to see what that turns up. If you have any other specific suggestions, it might be nice to talk further.
Yeah.
I have a question. So I have a question about your experimental design. So when participants chose cash out option, did you show them what would happen if they hadn't?
Not in this experiment, but we did do this in another study. So this is work by Tom Gibson, who was one of the people I mentioned. He was an honours student at Monash last year. Basically, it doesn't matter. So if you manipulate, sometimes we show people the counterfactual, sometimes we don't show people the counterfactual. It basically has no effect on their behaviour, which was a surprise to us because we thought it might.
It's just because we learn from the mistakes, like prediction error, kind of. That's the reason I was asking. Because when you're showing feedback, then it means participants are learning from their previous experience, and over the time they learn how they make their belief.
We haven't really looked at that just because there's not a strong learning component in this task. So I skipped over some details, but red and blue are correct in equal proportion of the time. There's no advantage to selecting one over the other. And so there's nothing really to learn in that sense. It would be interesting to look at that in a scenario where you could learn something.
Thanks.
Okay.
Yeah, thanks. A really interesting series of talks. I had a question for Daniel. I was just wondering if you ever study gambling in combination with like alcohol, like is there any influence of alcohol on this kind of behaviour that you see, or beyond that?
Sorry. No. Yeah, it sounds like the thing I try to get through the Monash ethics rather than the Uni Melbourne ethics. You're asking me about something I don't know very much about. Except to say that in general, what we know is that alcohol tends to exacerbate people's willingness to incur risk. So you might expect that to exacerbate this kind of effect, but otherwise I really don't have. There are some people in the room who might have something to say about some of this, but I'm not the right person to ask.
Question about user interfaces. So when people are gambling and they want to be in control, I'm guessing that, like if you're a Manchester United fan and you can get a user interface that has all the Manchester United logos and the colours and surreptitiously plays the theme song in the background, that's going to elevate you into a state where you think you're in control, but you're suddenly double, triple betting. Has anyone done any work along those lines?
Not exactly along those lines, though. It's definitely an interesting thought. One of the theories in this space that I actually wasn't aware of until a couple of weeks ago when a very bright, honest student brought it to my attention is something called the risk compensation theory, which is the idea that when you allow people to take more control or to feel as though they are more protected, they respond to that by taking less risk. Yep. And so you might expect that if seeing your favourite team's theme song made you feel more in control and happier, that you might compensate for that by taking more risk.
Now we've got a question here. Hi, my question is also for Daniel. I was wondering if you potentially noticed gender differences in the behaviour regarding the choices made, and then potentially if there's any really kind of work you've looked at in differences in behaviour and choices in regards to social learning theory, so particularly observational learning, in the context of, I guess, anecdotally, I've observed a lot of these products are marketed more towards a male audience, particularly leveraging the social aspects of it, like Bet with Mates. Yeah.
Really interesting question. Narrowly, when we look at, so we've mostly recruited sort of community samples for this stuff, and narrowly when we look at differences in those community samples, we don't see huge gender differences. I'm not totally sure how to interpret that, given that, as you say, that sports betting is a very gendered activity in Australia, and the promotion of some of this stuff is very much as well. And so we know that sort of being a young man is a set of demographic characteristics that puts you at greater risk of gambling-related harm. I don't know to what extent it's mediated by these specific features, and I don't really know enough about social learning to really comment intelligently on that.
Cool. Thank you.
Yeah, really interesting question. Thank you.
My question's for Kate. How did your experiment take into account lateral loss of function, so on one side, and in the future, do you think you'll take advantage of the post-stroke critical period of neuroplasticity?
Oh, very good question. So for the participants that were in this trial, they were very mild. So they were all independently able to walk. So yes, there was a lateralisation of their weakness after stroke, but it was quite mild. And then for taking into account that early period, so that's the trial that I worked on with Amy. A lot of the work that I do does look at that. So we do a lot of trials where we start within sort of the first few days post-stroke. There is different research, though, that comes out. It looks like the window is quite long, probably, 3 months. It's not necessarily days, because there's a lot that is going on after a stroke, from inflammation, swelling, other things like that. So we don't really know the optimal window, and then it is a little bit different for different impairments as well. So like a motor weakness seems to be a shorter optimal, versus language has a much longer window where you might see recovery. But I would just plug that it's not that recovery stops, it's just the slope that changes. So it's very steep early and then it's not as steep. So we do take it into account in other studies, but based on the observational stuff in Amy's study and that trial, they wanted to target that window of where it was maximally declining hippocampal volume.
So after 3 months, it's not too late?
No, definitely not. Definitely not. We have a trial, if I can ad-lib, we have a trial that we're running over there where we take people 3 months to 2 years post-stroke. And then there's other studies where they definitely see improvements months, years post-stroke. It's just likely that the conditions around training and learning are different. You might need a lot more exposure, so a lot more hours of therapy to be able to get the same effects that you might have seen earlier. But that's all stuff that we don't know in a lot of detail about stroke. Great question, thank you. Good question here.
Hi, I had a question for James. So I'm interested in this sort of micro offline learning that you mentioned, and I believe in what you presented, it was exclusively motor skills that you were looking at. Has there been work that tests if we see this in cognitive skills as well?
It's a very good question.
Sorry, I'm going to set you up for that, did I?
Very good question. The reason the paradigm really is well suited to something like motor sequence learning is that you can get this quite near real-time behavioural marker of the skill progression, lots of data points that then allow you to look at partitioning between the online and the offline period. That gets quite a bit harder with quite a lot of other task paradigms. So it could be quite challenging to think of a cognitive type task paradigm where you could get the sampling of behaviour and the rate of progression that would allow for that. There was a study that came out just this year focused on the neural markers of replay for the more working memory, or I'm going to have to look it up. But yes, so there's people starting to look at it from the neural point of view, would be what I'd say.
I also have a question for James. So it sounds like the best strategy to learn something, at least motor, is to take a break. But you mentioned the replay as well. And my question is, do people need to be aware that, like, are they actually replaying in their head, or are you referring to the ripples that are related to replay? And could you So in other words, if you asked people, if you probe people to remember, do they get better? Yeah.
So the neural evidence would suggest it's not conscious, deliberate rehearsal, that it's more of an unconscious, much more temporally sped up replay type activity. I can't actually talk about that study. People are starting to think about.
I'll take it for a drink.
Ideas of.
You're going back to your talk, yeah?
I'll just go so far to say that I think people are starting to think about ideas of like temporal memory reactivation, like probing and prompting.
A long time ago, I did a study looking at imagined movement and its movement-related potentials with real movement, and you could see similar brain activity. So people might be subconsciously running through that sequence and activating the supplementary motor area.
Yeah, I guess that's an open study to be done where we ask people on what strategies, but really they're just faced with a fixation cross, and my strong guess would be that most people will just enjoy taking a little break.
Yeah, right. Yeah, and their brains doing stuff. I'll just go to Dan.
I can probably shout, that's all right. Oh, yeah. My question's for James as well. You, in your review of past literature, including your own, you spoke about the task itself, and in every single one of those is 10 seconds of active and 10 seconds of offline. Do you have any sense of what happens when you vary those time periods? Is there a diminishing return on the offline gains?
So we're looking at a process that seems to be quite, rapid. It's those first few minutes maybe somewhat specific to sequential type tasks. There's unknowns for other types of learning that perhaps is a little less sequence related. There are studies where there are different lengths of task or rest duration, and the overall picture would say that it's somewhat insensitive to that, provided it's a sequence learning task.
When you say it's insensitive, you mean that?
Within bounds, it will make it more like 30 seconds for the task or 30 seconds or a minute for the rest. You can still see it. Where that disappears, on what timescale it would no longer be present. I don't think we know necessarily those boundaries, but yeah.
Could I provoke you to hypothesize a little bit? So if you've got a situation where, obviously you're varying the amount of time, do you think it's the number of offline opportunities you have so that, there's some maximal offline gain you can get, but you get lots of opportunities of that early on and you'll get maximal gains, but if you only give someone 2 or 3 opportunities with the same rest period, do you know what I mean? Does it? Yeah.
Yeah.
You get this additive component as well as the length of each section itself.
Yeah, we don't know. And of course, I'm not saying that we don't get benefits from actually practicing as well.
Yeah.
It's just a nice task paradigm that allows us to, at least to an extent, look at the contribution of what's going on when you're not actively practicing. It could be that you're even replaying things as you're doing it in the hippocampus, but being able to measure that with behaviour would be really, really hard, impossible. But by inserting the break periods, we get to look at the behavioural manifestation of what might be a replay type function.
Basically the same question. Is there a refractory period for motor learning where you've gone too long?
What do you mean by a refractory period for motor learning, where you can no longer learn anymore?
Yeah, if you don't go without breaks. But, well,
Sure.
obviously,
the elephant in the room for my talk is eventually fatigue might start to kick in, or fatigability. By keeping the task durations really short, we think the effects of fatigability type of processes are minimal. But yeah, if you had very long doing the task with very little break, then performance might decline because of fatigue, lapses of attention, you name it.
So this is my question for Kate. So I was one of the participants of Uplift thing, and basically I wanted to ask you about how. So I can see that you are having only ischemic stroke. So do you think that hemorrhagic stroke will be so basically, is it different in the outcome, and then also the exploratory outcome? What do you mean by neurodegenerative biomarker? Thank you.
Thank you. So only talking about the Pisces trial that we had, they focused on people with ischemic stroke because they were looking at the white matter hyperintensities, which is what happens in people who've had more ischemic strokes, so the blockage in the brain as opposed to the bleeds in the brain. So it's the mechanism behind what they were going after to try and measure is why they focused on ischemic strokes. Not all stroke trials do target one over the other. It really does depend on the time course of where they are. So a little bit in relation to your question, we've done it in walking where it was a large trial of 2,100 patients, and we saw that the time to independent mobilisation in people with a hemorrhagic stroke as opposed to an ischemic stroke is quite different early after stroke, but then it kind of meets up later after stroke. So it really depends where the study's happening as to whether we take people with ischemic and hemorrhagic or the mechanism that they're trying to tap into to improve from the recovery perspective. And then the second one was around one of the exploratory measures, wasn't it? Remind me which one you asked. I know I'm being a speechy on the side, aren't I, by making you the neurodegenerative. They collected a lot of bloods and biomarkers. That's very outside my scope as a physio. So those ones are coming down the pipeline. Thank you.
I have a question for Daniel. In relation to user control in gambling, have you explored the correlation of user behavior with the cash out feature in betting compared to, say, an amateur investor responding to the market?
The short answer is no, we haven't looked at that. The slightly longer answer is that has been suggested to us as a kind of a really interesting parallel between people have historically thought about gambling as a different kind of behavior to investing, but there's been a lot of changes in sort of investment apps and gambling apps that have sort of brought the two closer to one another, which is just my long way of saying it's a very good question. And I think there's likely to be some substance there, but we haven't really specifically looked at it.
I have a question for Daniel as well, and it's about when you said that the cash out option might be a gateway product for people to get into gambling. I was wondering if that was the case where people who are less impulsive or have a lower gambling severity score would, like because of that option, go into gambling, or is it the case that when they're put into this gambling context that they would be more likely to use the cash out option?
Yeah, it's a really good question. So what our results say is the latter. And what we are sort of speculating is possibly the former. So our results tell us that these are the people who like the cash out feature when we offer it to them experimentally. And then what we are speculating from that is that if we extrapolate that to kind of the population, you might imagine that those are the people who might find gambling as a product more enticing. We don't have evidence for the latter yet. It's a very speculative hypothesis. We are hoping to get some funding to run a longitudinal study where we sort of would look at how often people are using this feature and how much gambling harm they're having over time, to test whether sort of increasing rates of cashing out early on predict a sort of a different trajectory of gambling harm. But it's just a hypothesis at this point.
I have a question for Daniel. For your task, you allowed people to wager, but they weren't really putting a lot on the line. It wasn't even all the points that they had available. Mm-hmm. They had like 3,000 in the bank. You were allowing them to bet up to 50, I think. Mm-hmm. And it probably wasn't real money, or if it converted to real money, it probably wasn't much.
It amounts to 5 cents, something like that.
So a poker player, eventually, even a pretty rational individual, buying their time might have to go all in at some point. And maybe also people having a flutter, allocates them a certain amount that they can have for the day, and eventually they might go all in. Do you think that would change the behaviour patterns that you're looking at?
I think it would, and I think there's some really interesting possibilities to look at there. So one possibility is that we've sort of looked at the effects of cash out on betting behaviour, on individual betting decisions, but this is a task where there was a certain fixed number of bets that people had to make. They were doing the task for 20 or 25 minutes and then they finished up. But when people are placing bets using sports betting apps, they can bet all day if they want to. They can bet for 18 hours as long as they continue to have money in their account. And so one interesting possibility is that cash out might actually be a feature that has different properties in those real world environments because it allows people to kind of prolong their gambling sessions. So actually it might be a feature. So if you can't cash out and you have a run of bad luck, then you deplete your wallet and you have to either put more money in or stop betting. But if you can cash out, then what you have the option to do is to see when your bets are going bad, cash out of them and put the money into something else. And so what that would do is it would sort of reduce the variability of your payouts, prolong the duration of your gambling sessions. And what we know from other products, like poker machines, is that when you have those properties, the product becomes more and more harmful. So this is very much speculating beyond what we can say from our experiment, but there's reason to think that there might be effects of cash out on behaviour that are sort of notable and worthy of study that we kind of haven't got at in this context.
Hello. I have a question for James. Sorry, thank you. When you did the research, did you I don't really know much about stroke, but I guess stroke can affect different parts. I'm really sorry. I got really confused. Is that
make something up about
There's interesting stuff in that.
yeah, both. I'm so sorry. I got confused. But if you can both answer my question, that would be great. So I guess stroke can affect any part of the brain, but did you have any sort of link to what part of the brain is affected by the stroke and the result of your research? Like, was it really different?
All comers, if they've had an ischemic stroke, so the block, anyone like that, and those other caveats that I said of like if they were relatively independent and mobile before their stroke. So we do think, we know that what people are like beforehand can be part of the prognosis aspect. But no, we just took ischemics, people with ischemic. Acute stroke stuff does go by stroke location a little bit more, but the rehabilitation stuff is a little hard. I'm not saying that I don't think it probably does have a factor in recovery, but it's not there yet from that subgrouping perspective. We often subgroup on performance.
Yeah, another question for Kate. So you showed that there wasn't much of a difference in the hippocampus changes with the exercise intervention and more than yoga. I was wondering whether you or other groups have looked at that yoga in comparison to the incidental exercise that people do, walking and housework and so forth, and whether you looked at other areas as well.
Yes, not in this study. They haven't, and very little has been done because of the challenge with enrolling people in stroke research. We either have to have a waitlist control, so people will ultimately get the intervention. But for a study like this, where there's a temporal component, it becomes very hard because then you don't have people at the same time post-stroke. So at the end of this, you do have two interventions that were against each other because it was an active control and still an intervention. It would be interesting to know what it's like compared to doing not much at all, which is what most of our people with stroke are. So even doing 3 times a week of balance and stretching is a lot more than most people are. So that's why we take it as encouraging that it looks like doing something is better because they both didn't decline, but what intensity is best. There's lots of people who have looked at high intensity and moderate and low. It's a very varied landscape at the moment, particularly when you look at your physiological changes.
And so I have a question for Kate, which relates to intensity. So good timing.
I don't do a lot of exercise intervention studies as a physio.
It's a specific question related to the study.
Yeah.
So I'm wondering, it was described as a HIT protocol, and of course, rightly so. I think your group and other groups around the world doing these sorts of studies at the moment are progressing with caution. I'm wondering what the realised kind of actual intensity looked like. Was it in line with a definition of high-intensity exercise where heart rate max got up well over 80% of max, and perhaps you get him kicking over into anaerobic metabolism during those exercise bouts?
There's definitely periods where they do. One of the exercise physiologists is diving into all of that data now. So we know that they were per protocol in the number of minutes that they achieved. But what that profile looks like and whether certain ones led to better benefits and those sorts of things is not known. Because it was supervised by an exercise physiologist, there is a lot of encouragement that goes on and you can get people up to those high levels. However, some of the other work that we're doing is we actually give choice to the person to pick what intensity they would like to do. So we have, and that was a co-design project where people with stroke designed that and said they would like to have agency over it's moderate physical activity or vigorous physical activity. And so they can then self-select, and do you get better behaviour change with that side of it? And ultimately that's what you care about. We don't know all of that yet, but in this study they did fairly well, but that's the benefit of a randomised control trial is you do largely adhere quite well.
Great. Well, thank you so much to our three panellists. Let's all give them a round of applause. Thank you. And thank you all also for your wonderful questions. I'm going to hand over to Marta to close the proceedings.
Thanks, Catherine, and thanks for keeping us on time. You can't leave. I also have little presents, but I just want to say thanks again to Catherine for keeping us on time and making all of this engaging, and the speakers again and the audience. You've been amazing. I like that the questions came from different spots as well, so that's great. So I do have prezzies.
At last.
At last. Thanks so much. Thanks so much for your time. Okay, so you may sit in your spots now. Thank you. And I really just want to wrap up. So we're nearly done. I just want to say, again, thanks to all our speakers. Some came from Melbourne, some came from Monash, which is actually seems quite far. All our chairs, Iroise and Catherine and Sarah, thank you so much. Also the volunteers. So you saw that Daniel and Caleb, thank you so much for helping out at the registration desk and making sure everything ran so smoothly. And Sarah Davey, where is she? There she is. Thank you. Actually. She's an absolute star. She organized everything so well, prepared all the slides. Everything went so smoothly. Oh, and yeah, just thank you, really. And also the Melbourne business staff. Is that what the MBS stands for? Melbourne Business School. I'm not quite sure what they did, but probably were easy to work with. So thank you so much, all of you. And I think, are we doing a group photo? Can we do a group photo? Yeah? Okay, let's do a group photo. So maybe what we'll do is, those of you there, try to hurdle over there, and then Sarah can take a photo from here. Is that all right? Did I forget anything, Sarah? No, I think that's all. And then you're free to go.
Well, let's